So we should wait for others to join. Hello, am I audible?
Hello, yes, ma'am, you are audible, ma'am.
Okay, so we should start or wait for the others to join.
Ma'am, actually, Hemant is coming in 2 minutes. Then we can start.
Okay. I'm sharing my PPT. You let me know whether it is visible or not.
Good afternoon, madam.
Good afternoon, sir.
Sorry
Sir, I'm
for.
sharing my PPT. Please let me know whether it is visible or not.
Can you just have you shared? Yes, advanced signal processing techniques in EEG-based brain-computer interface.
Yes.
Yes, sir, perfectly visible.
So full screen is visible, right?
You have put it full screen. I think yes, it is there.
Okay. But then I won't be able to see you.
No issue, madam. What you are showing, that is more important.
Yeah.
Uh,
So
I,
we should wait for others to join, then we'll start.
so now I think we can start it because already.
Sir, actually your volume is very low. Either I should increase.
Just a minute. Hello.
Yes.
Yes, ma'am. Is it fine?
Yes, yes, sir.
Yes. So I think we can start. People will join it. Meanwhile, today actually it's Sunday, but actually within a time period that we have to conclude the workshop. That's why we
Yes, sir.
put on Monday, Sunday.
Yes.
So I welcome all the participants and our today's speaker, Dr. Gitanjali Srivastav. She has pursued her PhD under the supervision of Professor S.D. Joshi, IIT Delhi. I met the madam during our supervisor's farewell, and otherwise earlier I was aware that she had been doing some signal processing work and some multi-rate filter bank, etc. But now and I'm also very grateful that madam accepted the invitation for a session just before a few days when we had discussion that over it. That EEG can be a useful topic for the participants, especially from the industry perspective. They have been they have been recording their own data set and doing the different type of experiment. So without not taking now any further time, I hand over session to Gitanjali madam.
Okay. Thank you so much, sir, for inviting me. And good afternoon, everyone, the attendees, the host, and everyone. So let me share my PPT. Yes. So I hope it is visible. My PPT is visible. Please let me know whether my PPT is visible or not.
Yes, it is visible, madam.
Okay.
You are also well audible.
Okay. So let's start. So thank you very much for inviting me for the faculty development program, which is on advanced signal processing techniques. and you have so many sessions from so many professors like Professor S.D. Joshi from IIT Delhi, Gadre sir from IIT Bombay, and all other. So you, all of you must be having a good knowledge of advanced signal processing. So I'll be covering the application of it. So my topic will be advanced signal processing techniques in EEG-based brain-computer interface. And a brief introduction of myself. So I'm Dr. Gitanjali Srivastav, and I'm working as a neuro research scientist in Neurocognitive Artificial Intelligence and XR group in CDEC Delhi. And before that, I did my MTech and PhD from electrical department from IIT Delhi under Professor S.D. Joshi. And my main topic was in signal processing domain only. After that, I did a postdoc where I worked in a project which was in collaboration with the DRDO, and there I worked in the field of brain-computer interface, and that project was related to how we can drive or how can we means control exosuit exoskeleton from our brain signals. So that project was very nice and very exciting project. And after that, I joined CDEC Delhi here as a neuro research scientist. Here also I'm working in so many projects which are related to brain computer interface only. So I'll be covering these topics in this session only. So let's start with it. So first of all, what topics I'll be covering? First of all, we will know about our brain signals, and how and why to record EEG signals. The brain signals is EEG signals, so why we are recording EEG signals, and how we can extract EEG signal from our brain. So various tools are there for analyzing EEG signals, and then after that, I'll be covering standard electrode positions and reference systems, 1020 system, which is a standard head measurement and electrode placements in our EEG headset. After that, I'll be covering artifacts in EEG signal, because generally when we have recorded our EEG signal, then a lot of artifacts get induced in the main EEG signal. So how can we how can we process our EEG signal, denoise those signals? So I'll be covering that. And after that, how can we do EEG signal processing, feature engineering, and for that we will map it to from time domain to from other domain, so that will be transforming from one space to another space. And then I'll be covering in a short independent component analysis, which is very essential for feature engineering also, and as well as for artifact removal also. Then I'll be covering various case studies which are related to SSVP experiment, multimodal data analysis, and all that. So let's start with it. So first of all, here I'm saying I'll be covering advanced digital signal processing techniques in EEG-based BCI. So what is EEG-based BCI? So EEG is a brain signal, and it is a non-invasive method to record brain electrical activity via scalp electrodes. So BCI is brain-computer interface, and it is often called as mind-machine interface or direct neural networks, direct neural interface. So it is a technology to communicate human brain directly to computer or any communication device without any physical contact. So this is BCI, means we can directly communicate human brain to any computer or any communication device where we want it. So, okay, so the history starts with the Hans Berger discovery of electrical activity of human brain and in an EEG. So it has started from 1924, where the research began. And the research on BCI began on 1970 at the University of California, Los Angeles. And we can see many application of BCI. That is why we all are working in this field. So it has a huge application area. First of all, neuro rehabilitation, assistive technology, gaming, cognitive research. So like in assistive technology, it means like we are making exosuit exoskeleton which can assist a person or help a person in doing some task. And by gaming, I mean that neurofeedback games are there. Neurofeedback game in the sense that sometimes when we these games are for increasing our attention or increasing our cognitive power. So suppose if I'm doing some work and I'm not attentive, then how these neurofeedback can make me more attentive? Okay, so these neurofeedback games are designed according to that, so that the while playing these games, we will get feedback from the system to us, and so that we will get to know ki, okay, my attention was not so much, so I have to improve it, or I have to work hard, or I have to be more focused. So this will give me a feedback so that I will be more attentive, more focused. So a huge application is there which we'll cover. So what are BCI good for? Okay, so a brain computer interface, this is mainly used for communication for logged in person, as we can see in the picture also. Suppose a person is there, and in his one hand, he has one hand, but the muscles are damaged. Okay, so his brain signals are not are not able to give information to his muscles, EMG, to his muscle, so that he cannot close his fist or he cannot move his hand. But the hand is there, but the brain signals are not going over there. So here we can use BCI, like we can implant electrodes in this arm, which allows the healthy muscles to receive signals from the brain to close the hand or do any activity. And the other case is, suppose the person's hand is not there. So what we will do, there we have implanted the electrode in this arm, which will relay the signal from the brain to the robotic arm to close the hand. So this type of robotic arm, we use this type of signals in exosuit, exoskeleton also. So these are various applications where we can use BCI. So it is communication for logged in person, people with little muscle control, as I explained. So where the person is not enough, he has not enough control for EMG signals, or gaze tracking, or the people suffering from any disease like spinal injuries or anything where he cannot move his hand or any limb, then also we can use these type of signals. And these are very low bandwidth bit rate we have, brain computer interface, so we can easily use that. And in medical applications also, we have many applications, like for early diagnostic, like we have diseases like dyslexia, sleep disorders, peripheral neuropathy. So we can in advance we can predict whether that person is going to uh, has symptoms of these disease or not. So early diagnostics can be done, and accordingly the medications can be follow up. And in the case of rehabilitation also, like restoration of movement, combination with VR. So these are the special applications of BCI. Now coming to the BCI capabilities for military applications also. They are it comes under four type of interactions in military application. The type one is interaction with sensory cortices, where we have digital tongue or nose. So we share the sensory data, like human or other organisms sense sensory data incorporated into sensor fusion, and new senses, and expansion of new natural sensory perception beyond natural limits. Example, like we have augmented reality applications like AR, VR. So this is related to interaction with sensory cortex. And then we have interaction with motor cortex area. Its example is like exoskeleton limb operation or exosuit limb operations. So these are like augmented response, like that person is having his hand or limb, but he want assistant, he want uh, like a support. Like I was working in a project in my postdoc. So that project was sponsored by DRDO, and it was for soldiers. So our idea was like, when the soldiers are going in a field in a war, then they have to lift heavy boxes also. So can we develop exosuit or exoskeleton for them, which will assist them, which will help them in lifting heavy loads? although they have their hands, but when they are picking those load, then these assistants will extra force will be given to them, so they don't have to use their power. That exosuit will give them that assistance, and they can easily lift that weight or boxes, and they can run in the field. So this was the idea. So this is the one of the examples of interaction with motor cortex. So we have enhanced operated task performance and expand range of manual tasks that the operator may be considered competent to perform. So the third type of interaction is the interaction related to cognition. So cognition enhancement beyond natural ability. So like synthetic telepathy or brain to brain interface we have. So communication of complex concepts such as operators intent to autonomous system. So this will come under this. And the last type is the type 4 is interaction with autonomous nervous system plus medical intervention, where we use it in our medical field. So like mimic psychological states typically associated, regulate awake sleep states. We can record the EEG signal of a person when he is sleeping or when he is awake also. So different applications are there, and monitoring operators medical conditions also. So this will be used in medical field. So let's start with know your brain, because the EEG signal which we are talking about, the BCI, brain computer interface, which we are talking about, this is all related to our brain only. So first of all, we should know about our brain. So, as we know, the brain is a complex organ that controls our thought, memory, emotion, touch, motor skill, vision, breathing, temperature, hunger, and every process that can regulate our body. Every process which we can think of is regulated by our brain only. It sends and receives chemical and electrical signals throughout our body. And different signals control different processes, and our brain interprets each. So, if broadly talking about, we can say our brain is divided into two parts, the cerebral cortex and the subcortical regions. And our research mainly focus on cerebral cortex systems only, that is the outer part of our brain. And if we are talking about the outer part of our brain, then we can also divide it into two hemispheres, like the left hemisphere and the right hemisphere. So these are functional areas of our cerebral cortex areas. So like we have different regions in our brain which are responsible for different activities, and these regions are called, they have specific names like primary motor cortex area, primary sensor cortex area, somatic sensor, visual association area, which are responsible when we are looking or watching anything. Then we have visual cortex area, auditory cortex area. These are normally at the temporal region of our brain. Then auditory association area, Broca's area, prefrontal cortex areas. So these are the different regions of our brain, and different activities get activated through these areas only. So here we can see broadly our brain is divided into four lobes: frontal lobe, parietal lobe, occipital lobe, and temporal lobe. So temporal lobe, this part is just above our ears. This occipital lobe is at the back of our brain, and parietal lobe is over at the center of the area, and the frontal lobe is at the area above our forehead. And here in this picture you can see that the different region of our brain corresponds to different activities. Like this prefrontal area, we can see this is responsible for our attention, planning, decision making. So if any task, we are coming across any task or anything, then if it is related to our decision making, attention, or planning, then this part of our brain gets activated. So if we are recording our EEG signal, then we can mainly focus on these part of the, if we will place our electrodes over these areas, so we will find a good signal over here. We can see the activation, how our brain is responding. If any task is related to movement, like we are moving, we are lifting our hand, we are walking, or we are doing anything. Then the motor cortex area, this movement, this part is responsible for that. For speech, smell, we have this region. For hearing, like for touch, taste, we have parietal lobes over here. For vision, we have occipital lobe, which is at the back of our brain, this head, above the neck. So this part is called as occipital lobe. You can see over here in this is in pink color. So this is responsible for our vision. So any task which is related to vision, by watching, or some, where we have to focus, where we have to look, then this occipital lobe, the electrodes which are on occipital lobe, they will give us a good activation over there. So we can do our signal processing and all our processing over these electrodes, over these signals only. Then we will get good results rather than focusing on other signal which are related to touch or taste. So, and this part is related to memory processing, movement coordination also. So we can see. So our brain's cerebral cortex is the outermost layer that gives the brain its characteristic wrinkly appearance. The cerebral cortex is divided lengthways into two cerebral hemispheres connected by the corpus callosum. And traditionally, each of the hemisphere, like the left and the right, has been divided into four lobes, as I said: frontal lobe, parietal lobe, occipital lobe, and the temporal lobe. Moving to the next slide. So let's decode our brain. So we have divided our brain into mainly four lobes. That is the frontal lobe, parietal lobe, temporal, and occipital lobe. And these lobes have different locations and their primary functions. We will discuss over here. So frontal lobe, this is the front part of the brain, and this is mainly responsible for thinking, decision making, problem solving, planning, emotions, voluntary movement, or motor cortex. Motor cortex related to activities related to motor movement. Then we come to parietal lobe. We can see over here this yellow region. So this is the top middle part of the brain over here, and this is responsible for sensory processing like touch, temperature, pain, spatial awareness, hand-eye coordination. When we need hand-eye coordination when we are doing any activity, then this parietal lobe, the electrodes or the signals over this region will give the accurate information about the brain activation, if we'll take that. And the temporal part, this is the side of the brain near the ears. So this is responsible for hearing, language processing, memory, emotion regulations. So in our like when we do experiments or we design some tasks, then we make paradigm, and in those paradigms we uh, we have certain task for the subject, for the participant. Like first he has to close his eye for a few seconds, then for he has to open his eye for a few seconds. This is for relaxation. And then after that we start our task. Like he when the beep sound has come, then he has to perform this activity. After that 5 or 30 seconds, the next beep sound will come, then only he has to respond. So these beep sound, he will be attentive in listening those beep sound, and these beep sound which he is listening, this will give activation over the temporal lobe, the sides of the brain. So if we'll see the signal over these areas, then we will get the activation. Whenever the beep sound has come, we know that at what time we have given the beep sound. So with corresponding to that, we can see at the EEG signal that the electrodes which are at the temporal lobe, they gets activated, and these signals will have these the signals which are at these electrodes will have high amplitude rather than the other electrodes. Then we have occipital lobe. This is at the back of the brain, and this is responsible for vision processing, object recognition, color perception. So we have some task, like I will later I will show you. So the task, like if we very famous, very popular task is that the odd ball paradigm. task is there. There you will see that the like the participants will be will sit in front of the screen, and in that screen he will see that a one circle is coming or one ball is coming, and that will be of, suppose that will be of blue color, so it will come in blue color. Then again one ball will come blue, 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 and suddenly the color of the ball will change. It will be red color or any other color. So the brain gets attentive, the brain gets stimulus over there. So this so we can see when we see a red ball, then our occipital lobe, the electrodes or the over there, they will have a very high amplitude, irrespective of the other electrodes. You will see that. So different tasks are responsible for different functions, and according to their functions, different electrode, different lobes will be get activated. So in our research analysis, we should focus on these type of things only, which this task belong to what type of function, and this function will initiate which part of the lobe, which part, which electrodes. So we should focus on that only rather than considering all the electrodes. Because when we are considering, we have EEG headset of 64 electrodes also, 32 electrodes also. So so if we are considering 64 electrodes, so we have 64 diamond, 64 cross. Suppose 5-minute data is there, so five and sampling frequency is 500. So you will have a, two-dimensional matrix which is of 64 cross 2500 this data only for 5 seconds. So this data, this huge amount of data, why to handle this huge amount of data? This will increase our data redundancy also, and this will decrease our accuracy also. And when we are translating it to, we are doing feature engineering, or we are making AI model for that, or machine learning model for that, then it will take a lot of computation time, as well as the accuracy will not be that much correct also. So rather than focusing on all electrode, we should focus only on the specific electrodes which are responsible for those functions only. Now coming to types of BCI systems. So mainly there are three types of BCI system which we have. First one is non-invasive, then second is semi-invasive, and third one is invasive. So what is non-invasive method? Actually, mainly we are using non-invasive only. All the research analysis are done on non-invasive BCI systems only, where the sensors are placed on the scalp, on the scalp to measure the electrical potentials produced by the brain EEG signal or the magnetic field MEG signals. So this is like we have this EEG cap and we have just wore it, uh, and nothing surgery is over there or any electrode we are placing it inside our brain. This is not like that. So this is non-invasive method. Then we have semi-invasive method. This is this is where the electrodes are placed on the exposed surface of the brain, which is ECOG. These signals are called ECOG, and it is called semi-invasive, but it still requires a craniotomy to implant the electrodes. Electrodes are implanted inside it, and for this reason, it is used only when surgery is necessary for medical reason, like we have epilepsy or another. So where the these surgery has to be done, and these electrodes are placed over there only. Then we have invasive method. This is totally surgery based. So they are implanted directly into our brain during neurosurgery, and the micro electrodes are placed directly into the cortex, measuring the activity of one or more neurons over there. So we will not go deep into the semi-invasive and invasive methods. We will only research about the non-invasive methods. And like I said, we have EEG cap or EEG headset where we just wore it. But and we have different types of EEG headset also. Like in different many companies are there, like Emotive, Brain Products, Wingen, Neuralink. Many headset with many companies are there, and they have this EEG electrode. The electrodes, they are of three types. Like first we have saline based, the water based EEG headset. Other one is gel based EEG headset, and third type is dry electrode headset. So if I have used all these three headsets in my project. So out of all these three headsets, I would say ki the signal quality of gel based EEG headset is very high. Means very better as compared to EEG saline based or dry based electrodes. If I'm talking about, actually, if we have wet electrode EEG headset or EEG cap, that means that the electrodes are directly connected to our brain, either through the gel, which is a conductive material, or through saline water. But in the case of dry, they are not directly connected. The electrodes are just touching our scalp. So the signal quality in case of dry electrodes, we get less signal quality as compared to gel based. But there are also many technology and many uh, advanced technology are coming. So we are getting good signal quality in dry electrode headset system also. And if I compare saline based and gel based EEG headset, then I would say gel based EEG headsets are better than saline based, because when I was using saline based EEG headset, then we found that the water they get dried in 15 or 20 minutes. So when we have a experiment of 1 hour or 2 hour, then this saline based EEG headset will not work. Otherwise we have to means we have to put saline over again and again, so it will disturb our experiment. So better go for gel based EEG headset. Mm, so BCI can be applied to variety of tasks like neurofeedback. This is neurofeedback, which where we are getting feedback from the game, neurofeedback games, to us, so that we can become more focused, more attentive. Then we have restoring motor functions to paralyzed patients, and this also allows us in communication with logged in patients and improving sensory processing also. So we talked about BCI, and the signal which we will record from our brain will be EEG signal. So what is EEG signal? So EEG signal is a technique used to measure the brain's electrical activity through electrodes placed on the scalp, and these electrodes detect voltage fluctuations caused by the activity of the neurons and provide a real-time representation of brain signals. So typically the electrodes are attached using a sticky gel, but research is going on, as I said, on dry electrodes also, because they are very convenient to use. The advantage of dry electrode is that they are very convenient to use. Means we just have to wear that headset and just make a arrangement that is touch the scalp and nothing else. But in the case of gel based, we have to put gel in the electrodes and in our scalp. So the subject, the participants get irritated because the gel, we have to the gel gets attached to their hair. So it is very not very convenient. So if I talk about the convenience point of view, then dry electrodes are very good. And the system requires a physical connection to an EEG machine to accurately record and process the signals. So EEG is widely used in neuroscience, medical diagnostic, and brain-computer interfaces to study brain functions and detect specific patterns like event-related potentials. So we have various system and applications like P300 speller, where EEG helps convert neural signal into commands. We will see it in later slides how this is being done. So through P300 speller, we can convert our neural signals into commands, and it is a precision and versatility make it an essential tool for understanding and utilizing our brain activity. Coming to a little EEG background, so in 1875, Richard Catton discovered electrical properties of exposed cerebral hemisphere of rabbits and monkeys, and from there it started. In 1924, the German psychiatrist Hans Berger discovered alpha waves in humans and invented the term this EEG. In 1950, the Walter Gray Walter developed EEG topography, that is a mapping electrical activity of the brain. And from there, we are developing so many things using brain computer interface. Now, after EEG, we will see from signal processing point of view, we will say that as we know, all brains, all real-time signals, whether it is acoustic signal, speech signal, EEG signal, ECG signal, or that avalanche signals, anything that water signals, any signal which we can think about are non-stationary signals. In real time, they are non-stationary signals. So similarly, EEG also a non-stationary signal. And when we are saying it is a non-stationary signal, we mean that the characteristic of these signals are varying with time. The statistical characteristics of these EEG signals are varying with time. So we cannot apply like if we want to do some transformation or some feature engineering, or we want to apply Fourier transform on EEG signal, then we cannot just take a 10-minute data of EEG signal and can apply Fourier transform over there. No, we cannot do that, because Fourier transform is for stationary type of signal. These formulas are for stationary signal. So first we have to assume it. We have to make these EEG signal convert it into stationary signal. How we can convert them into stationary signal? Just like we do in our speech signals, audio processing, what we do, we take a small window, small time window of these signal, and we assume that these signals are stationary, their characteristics are not changing in those short windows, and then we apply Fourier transform on those windows, and then we do our further analysis, whether it's power spectral density calculation or anything. So that must be taken into account. Otherwise, the results will not be correct if we are taking the whole EEG signal and applying Fourier transform. So this is the thing. And so we assume these signals to be quasi-stationary, like we assume them to be a stationary for short period of time, and then we do our transformation and analysis. Coming inside the EEG signal, since EEG signal, they are, if I talk about their frequency, then their frequency range from 0 to 250 hertz, or 100 hertz. But the most useful frequency component which are essential, we generally talk about from 0 hertz to um, 50 hertz or 60 hertz signal only. So in that range, we have divided the EEG signals in five brain wave, five brain waves categories. So let's start with that. So brain disorders are often diagnosed by analyzing EEG signal through visual inspection, like the doctors or the experts, they can do it through visual inspection only. We, the engineers or researchers, we are not that much expert enough that just by looking at the EEG signal or their rhythm, we can say, okay, that person is suffering from that disease, or he is having so and so disease. We cannot say that, but the expert and doctors can say that. Clinical experts recognize different brain rhythms and their manifestations in EEG signals. Like they divide the EEG signal into different bands, and then they analyze those signals in that bands only. So if we are talking about the characteristics, then EEG signal characteristics, this includes amplitude and frequency and change with states such as wakefulness and sleep. So when the person is not very active, he is sleeping, and you will see his EEG is mainly is coming in the delta and theta. When it's the low frequency components, you will see low frequency components over there. So amplitude in other frequency components will be very less, and the amplitude in these low frequency bands will be very high. And when the person is very attentive, very wakeful, then we will see high amplitude in higher frequency ranges. So through these waveforms also, by visual inspection we can say whether the person was attentive or not. Okay, and the wave character and these wave characteristic which we have shown you is also vary with the age. So there are five major brain waves exist, categorized based on frequency. So first is delta band. This is from zero point 0.5 to 4 hertz. Then we have theta band. This is 4 to 8 hertz. Alpha band. This is 8 to 13 hertz. Beta. This is from 13 to 32 hertz. And the gamma is from 32 to 100 hertz. So coming to in details, so first of all, the delta waves, these are from zero point 0.5 to 4 hertz. So this is mainly associated with deep sleep, but can also appear in wakefulness. So when the person is in deep sleep, then these delta waves are going on. So we will see the amplitudes in delta waves only and not in other frequency, but sometimes we can see also in wakefulness also. And this can be confused with the artifacts also from large muscle, like we have neck and jaw artifacts. When we record EEG signal, then we have multiple artifacts are there, like the baseline wandering is there, which is normally at the low frequency component. Then we have some muscle artifacts which are at high frequency components. Then we have some power line interference also, which is exactly at the 50 hertz or the 60 hertz. So these delta waves, these are sometimes get confused with the artifacts of low baseline wandering type of artifacts. So delta waves originate deep within the brain and are highly attenuated while passing through the skull. So signal analysis can distinguish genuine delta responses from the motion artifacts. So if we want to distinguish these data responses from motion artifacts, then we have to do some signal analysis things, which we do. Now our next is theta wave. So theta wave, they may originate from the thalamus and appear as a consciousness shift towards drowsiness. So okay, so it is like a little better than drowsiness. And this is associated with access to unconscious material, creative inspiration, or deep meditation. So when a person is in deep meditation, we can see their theta waves, you know. And in our research, we when we when the research is related to attention, then if we want to compare whether this person is more attentive or that person is more attentive, then we see their theta wave ratio, the power, uh, you know uh, the power in the theta wave or the alpha wave, whose power spectral density or power the amplitude was high, then that means that person was more attentive, more focused. So these are helpful in that also. And these theta waves often accompanies other frequency waves reflecting different arousal levels. And their significance in infancy, they are significant in infancy and childhood, but then normal in waking adults. Like in the adults, if we are saying theta waves, normally we see theta when the person is attentive doing work, an adult is doing that, and he's getting the high amplitude in theta wave, then some abnormalities is there. Otherwise, we will see theta waves normally in the infants and the babies only. So it is used in maturational and emotional studies to track brain development, how the brain has been developed. So these waves are used in that. Then we have alpha waves. So they are mainly found in the posterior half of the head, particularly in the occipital lobe, and commonly appears as a sinusoidal wave, but may take other shapes in rare cases also. And this indicates relaxed awareness without attention or concentration. So these are the most prominent rhythm in brain activity, sometimes extending the into the beta range also. So from 8 to 13 hertz or more than 13 hertz, these frequency components are very essential when we are doing any analysis. So alpha waves diminish when the eyes open or in response to unfamiliar sound, anxiety or concentrations. And the exception case is the Albert Einstein reportedly maintained an alpha state while solving complex problems. Then coming to beta waves, so they are associated with active thinking, attention, and problem solving. A high level beta wave is observed in panic state. When the person is in panic state, or he is very attentive, or he is solving his problem, then we will see these beta waves found primarily in the frontal and central brain regions, like the frontal uh, for this area and the central region. The central region is mainly related to the motor cortex area. So when we are doing anything, we are moving our limbs, we are doing any activity, then our motor cortex area gets activated. So we will see these beta waves over there. And then coming to central beta rhythms, they relate to the mu rhythm and can be blocked by the motor activity. So, and their amplitude is usually from below 30 microvolt. When coming to beta waves, these beta waves may be enhanced near bone defects or tumor regions also. From alpha, theta, beta, we have next our wave is gamma waves. These are above 40 hertz, typically up to 45 hertz. So they are low amplitude and very rare in occurrence. So we will not find generally. So their detection can confirm certain brain disease. If this wave is very prominent in any person, then it means that the person is having some brain disease. This correlates with high EEG frequency. We can see high EEG frequency because it is coming at 30 hertz or above that, and this is their cerebral blood flow, oxygen, and glucose uptake. So they are used for ERS studies like event-related synchronization, where we are studying those, and this can be used for that. And they are helpful in mapping motor movements like right and left index finger movement, toe movement, and tongue activity. And after 45 hertz, we generally for research or for any activity, we take EEG signal from 0 to 50 hertz or 40 hertz only, and above that we call it a high frequency wave. So they are found in cerebral structure of animals, and they are not significant in clinical neurophysiology at present. So coming to in concrete, we can say we can compare the EEG band. So here we can see delta band. They are low frequency components. So we can see this low frequency waveform. Theta, they are 4 to 7 hertz. So greater than delta, but less than alpha. And this is waveform of alpha band, beta, and these are gamma are very high frequency components of EEG. So we can see in its waveform also. So in concrete, the delta bands, their frequency is up to 4 hertz, and their location is frontally in adults, posteriorly in children, and they are high. These are high amplitude waves. Means the amplitude of these signals are very high. So that is why we say ki sometimes it gets confused with the artifact also. When we have baseline wandering, so they are also these artifacts are also very high amplitude, and they come at low frequency. So normally these delta waves are in adults when they are in slow wave sleep, and they are found in babies and has been found during some continuous attention task also. In theta, these are 4 to 8 hertz, and they are found in location not related to task at hand, and they are found in young children, drowsiness or arousal in older children and adults, fiddling, and they are associated with inhibition of elicitatory response, and has been found in a spike in situations where a person is actively trying to repress a response or actions. Then we have alpha 8 to 13. They are posterior range of the region of the head, both sides, higher in amplitude or non-dominant sides, and they are found when the person is relaxed or reflecting, closing his eyes, and are also associated with inhibition controls, mainly with the purpose of timing inhibitory activity in different locations across the brain. And beta, they are generally greater than 13 hours. They are found at both sides of our brain, symmetrical. These are symmetrical distribution, whether it's in left hemisphere or right hemisphere, and most evidently they are in frontal part of the brain. They are when we are very alert, working, active, busy, anxious, thinking, active concentration, then we mainly find beta waves. Then we have higher frequency component like gamma and mu. So they are related to that. So this is overview of overall EEG processing and its applications in conflicts. Like we have EEG signal. This is our EEG signal. We have acquired this EEG signal, or we have recorded this EEG signal from the participant. Then what we can do? We will do its real-time applications. So its real-time applications are like neurofeedback. We will use it in neurofeedback. Some BCI application like in speller, robotics, or movement of our wheelchair, like a person with the physically disabled people who cannot move their limbs and they're sitting on wheelchair, then they can, through their brain signals, they can move their wheelchair, or they can do move their wheelchair left, forward, backward. They can stop their wheelchair. So these type of activities can be done through BCI applications. Then we have computer aided automatic disorder detection also, like in the case of diseases. So these are real time application of EEG signal. And what are the challenges which we face in while dealing with EEG signal? The challenges are like, first of all is EEG denoising. The EEG get contaminated with a lot of noises. When you will record EEG signal, then the subject has to, sit straight. If the task is related to uh, sitting, when the person is not walking, even if he is sitting, then he cannot move his neck. If he is moving his neck, then one artifact will get induced in the EEG signal. If he's blinking his eyes, if he's moving his mouth, or he's clenching, then these type of noises, which we know this type of noises will get induced, but there are other noises also which we don't know, which induced into our main EEG signal. So EEG denoising is very essential, and this is a channel. For that, we have different signal processing methods are there, like wavelets is there, independent component analysis is there, regression, EMD, VMD, empirical mode decomposition, variable mode decomposition, and adaptive filtering is there. So we have these methods for EEG denoising. Now, what the challenge is what the challenge is for offline analysis. Like once we have recorded our EEG signal, we have saved these EEG signals, and we have that in our system. Then we can do all pre-processing things. We can apply our formula over there, and we can clean those signals. But the problem, real time problem is this: when we have real time application, when like the EEG is coming, and that EEG, that brain signal will give command to device or to any wheelchair on that. So for that type of application, we do not have that much of time. So our algorithms should be robust enough. It should take less computation time, and it should be very accurate also. So how can we do these denoising? Like ICA, we can clean EEG signal through independent component analysis, but this ICA takes lot of time while cleaning any signals. So we have to focus on those EEG signals, on all those methods which can do our real-time EEG denoising. And also, like these methods, wavelet, ICA, EMD, all these methods are for they are applied for block of data, means for uh, they are for quasi stationary signals. Uh, But when our EEG signal is coming data point by point, then what algorithm should we use for that? for denoising of EEG signals. So these are all research areas where we can focus on. And other challenge is EEG source localization, means which part of the brain is getting activated in this type of task. So I should focus on which electrode, which part of the brain. This is also a very challenging problem. So there are many Python libraries and many tools are there where we can see this source localization. Like in Python, we have MNE library, DSPM libraries, where we can see the source localization. Then in MATLAB, we have Loretta over there. There we can see the which of the where we can see the source localization. Means visually we can see, okay, this part of the brain is getting activated, this is getting high power spectral density, and the other part of the brain is getting low power spectral density. So it means with this part is getting activated, the motor cortex area is getting activated, occipital lobe is not getting activated. So these are the challenges which we have to see, which we have to focus before actually designing any our overall methodology. Can we take a short break?
Yes, yes, Arnab, definitely.
Let's take a short break
Ah,
for 5
yes,
minutes.
yes, absolutely. So we can have a see break, or we will meet at two uh, around 2:40.
ठीक है.
10 minutes break. We got
Okay, sir. Let's start, sir.
Okay, ma'am. So yeah, we can restart the session. So to the participants, if you have question, you can type the question in the chat box so that because see, session will be successful when you will also able to absorb the content, see what has been delivered. So see, without you can type it, and as soon as there will be break or we will have the time, the question will be taken. So session, madam.
You can see my slides now, sir.
Yes, now it's visible. Now you are showing general structure of BCI.
Yes, yes, yes. So now coming to general structure of BCI. So general structure is like we have over here, the participant is here. We are acquiring his signals. Then we will do some pre-processing, like we will do, like we have the signal acquisition method, like recording of the EEG signal. And after getting the raw EEG signal, we will do some pre-processing. Pre-processing in the sense that we will remove some artifacts from it. Then we will do artifact removal. The artifacts can be many type, like we know specific artifacts like baseline wandering, power line interference, high frequency component is there, or where the eye blink artifact is there, muscle artifact is there. When the if the person is breathing very loudly, then that heart rate artifact is there. So all these artifacts are there in the EEG signal. um, uh, so we have to pre-process our EEG signal first, uh, and then after pre-processing, then we will do some feature extraction. We will extract the useful feature from those EEG signal. Then we'll do our classification. It will be our depending on our application, and then we will have some application interface, whether we want a spelling device, neuroprosthetic wheelchair, or neurofeedback game, or exoskeleton, or anything which will be the application interface device. So we will connect those things with this. This is a classification. If the model is classification model, then it will come classification. And if we have a regression type of model, like when we want a continuous value from our EEG, like in one of my projects I was working, where we wanted like how this hand is moving, this elbow angle at each and every every point. I do not need the classification whether the hand was up or down, not this type of, at what angle this hand was moving, with what angle. So these are angle, these are continuous value. So this comes under regression problem. So from through EEG, I exactly wanted the angle. So this continuous value will come over here regression, and this will be fed into our application interface, our exosuit total prototype, and then it will go, and this the angles will be fed into those, and accordingly the control part, the motor will provide that assist, that much force, that much assistant to the device. And if it is related to a neurofeedback system or SSVP, then these whatever is fed into the device will again give a feedback to the person, and according to those feedback, the person will again perform. So this feedback comes when we have some neurofeedback type of applications, where we want to increase his attention or cognitive power, then the feedback is given to the person. So this is the overall BCI scheme. Means we have here, we have recording, brain activity, recording device is there, then the preprocessor is there, decoder will be there, and external device will be there. So this is total the instruments or the hardware which will be required over here. This is one of the examples of role of BCI neurocognitive rehabilitation. This is just a pictorial diagram of that. Here you can see that this is a participant, and and this is a non-invasive approach. Means we have just placed a EEG headset into that person, and we are getting EEG signals into our system. So EEG recording is going on. After getting these EEG signals, we will pre-process it. We will extract useful information, and that feature will be fed into our machine learning model or AI model, and that will provide a control signal which will be given to our different assistive devices like assistive rehabilitating BCI system, or that will provide alternative communication and control methods for permanent disability or restored impaired brain functions by leveraging neuroplasticity. So this is one of the examples of neurocognitive rehabilitation. So for analyzing EEG signals, we have various tools are there which are available. So let's discuss about those. So EEG signals analysis requires specialized tools for pre-processing, artifact removal, feature extraction, and for visualization. So first one is Python-based libraries. There are a lot of Python-based libraries are there, which give us very powerful tools for EEG analysis. The first one is MNI Python. Their purpose is EEG, EMEG pre-processing, visualization, and analysis. So its features are like we have raw data loading. The data which we have recorded through our EEG devices, normally it is in the form of VHDR or EDF or FIF form. So we load those data into our MNE Python library, and then we filter. It will filter we can do filtering, artifact removal, like ICA can be done through that, and then we can do epoching and event detection also. Suppose we have some paradigm where we have given certain tasks to the participant to do that. Suppose we have shown him a picture and then we have asked him to respond according to that picture, or we have given him some questions or some mathematical task is given to him, and then he has to respond either by pressing the key or by pressing any button. So uh, at what time he has pressed that that pressed that button, so the exact timing. So we want that epoching of those events and detecting those events. So all these things can be done in MNE Python. And the source localization and connectivity analysis, it also provides statistical analysis. Also source localization, by means of source localization we means that we can see which part of the brain gets activated. We can see the picture of those in many Python libraries. Then the other library is EEG Lab. This is very powerful library which we have. Normally it is MATLAB based, but now it has been accessible by Python also. So this EEG Lab, through EEG Lab, we can do EEG pre-processing, ICA-based artifact removal, time frequency analysis also. And it has many features has been introduced in EEG Lab, like graphical user interface for easy data exploration, ICA for noise removal, and custom plugin support for extended functionality. If we want further analysis like VRD or any other of functionality which are not in the base model, then we can do it through custom plugin also through EEG Lab. Then we have NeuroKit 2, and it is easy to use toolkit for biosignal analysis, including EEG, and it is also used for pre-processing like filtering, baseline correction, power spectral analysis, event-related potential computations. And then we have PyEEG also. This is for feature extraction of EEG signal. This computes entropy, fractal dimensions, and other nonlinear measures, and are useful for EEG-based classification tasks. Those were Python-based libraries, and then we have MATLAB-based tools also. So EEG Lab, I'm saying EEG Lab is very powerful tool that is inbuilt in MATLAB, which we can use. So it is used for EEG pre-processing and ICA-based artifact rejections, and this is widely used for ERP analysis, frequency analysis, and visualization. Then we have Field Trip. This is for source localization. And for time frequency analysis, connectivity measures. Then we have brainstorm. In brainstorm we have S Lorita, Lorita. So this is used for source localization and also for function connectivity analysis. So now coming to how to record EEG. We have seen that through these are the tools through which we can analyze our EEG signals. Now coming to the first part of our thing, that is the recording EEG signal. Once we have recorded EEG signal, then we will do its pre-processing, feature extraction, and other thing. So first is how to record EEG signal. So this is a participant whose EEG signal, whose brain signals we want to record. So we have he has wore his EEG cap. There will be one amplifier which will convert these signals into digital form, and this will be fed into our this system. Now, how we record EEG signal or why we want to record. Either it will be some experiment based, paradigm based. We have different types of paradigm or experiment for which we want to do those experiments. Like suppose I was talking about the project of yeah, like it was related to exosuit. So there we made the paradigm. We made a certain paradigm where we shown him that okay, this is the start time, and when you will see uh, when you will see this picture of the bicep curl or the hand, then you will start doing this bicep curl, and for 4 seconds you will do one bicep curl, and when the stop will come, then you will stop. When the start signal will come, then you will start. So these are stimulus which are given to the participants, which are shown to the participant, and he has to perform according to that. And how he, and while performing those tasks, we will record his EEG signal. So we can see when this task was given to him, or when stimulus was given to him, and he was doing this task, then how his brain was performing. So this is how we record EEG signals. So we have recorded EEG signals. On the screen we will see these here in the columns we will see the electrodes. If it is a 32 channel EEG system or 64 channel EEG system, then we will see those 64 EEG signals, and this x-axis will be the time corresponding to the time. So throughout as the time pass on, how the signals are varying. So this is example of a printout of an EEG recording. This is a raw data. And if we have given him a certain stimulus, then through these EEG signals, we can do some clean, and we will clean and average that data, and we will do these ERP related analysis like P1, N1, P2, and 400. These are some ERP related epoch related neural correlates like P100 and 400. So I will discuss these P2 and 100 and 400 in my later slides in detail. So from these recorded EEG signal, we will do these activities. So here in this picture, we can see the person is wearing EEG cap. These are electrodes, and here we have EEG reading. So this is just one picture which I have shown over here. This is of my lab when I was in IIT. So here on the screen, we are showing him the paradigm, what he has to do, and this EEG cap which he is wearing, it is saline based. So at that time, I was using saline based EEG cap. So we have to put that saline through the dropper in these electrodes so that it gets connected to the scalp. And this is 32 channel EEG cap. So 32 signals were there. And this wire is connected to live M, which he has kept in his pocket. So, and he is performing the task. Okay, so what the task was given, when a certain paradigm is shown that this is the signal, now you have to do bicep curl, and this this task was related to, if he is holding some weight, then he has to do some bicep curl. So he is holding that weight and he is doing this task. So this is was recording of the signal. Now coming to we have to bore EEG cap. Now, how we should place EG cap on a person head, because everyone's head structure is different. Some some has the dimension of some people is is more or high as compared to other person. Some has big head, some has small head. So, but we have one EEG cap. So how should we place that EEG cap so that we can get standard data from those people? So there is a standard electrode position and reference system which we should follow while placing those EEG cap into that person. So standardized placement system of EEG electrode established in 1958 by Ed Jasper, a Canadian psychologist. So it is meant to facilitate communication and comparison of results, because those occipital lobe, if we are referring, this is the occipital lobe and these are the signals which we got corresponding to occipital lobe, but we have not placed our electrode on those occipital lobe, then how can we compare our results, or how can we say, okay, this is my result and I am getting this and this accuracy? So that will be wrong. That is why there is a standard electrode position system, reference system, which we should follow. So this provides for an adequate coverage of all parts of the head or brain. So this is called as number 10-20 system. Here, the number 10 and 20 refer to the distance between the adjacent electrodes. So you can see over here, this is a side. This is a side view of a person, and this is the top view of the person. So here in the top view, you can see this is a nasal nose of that person, and this is the back side of the brain. Okay, so this is top view of the brain, and this will be his left hemisphere, and this is his right hemisphere. So it is usually what we do. We this 10-20, this means that from this nasal point, then nasal point, we have to take 10% of the distance. The first electrode will be placed, and from this inion also, the 10% of the total distance, this will be our the electrode at the occipital lobe. After this, then all the electrodes will be at a distance of 20-20-20%. So this is how the EG cap is made, that the first this from the nascent and from the Indian, the first electrodes will be 10% of the total distance, and other electrodes will be 20% at the distance of 20%. And through the top view also we can see like this, that this is 10%, and then all other electrodes are at 20% at the distance. And this is the terminology which we used in our EEG headset. Each electrode has a letter to identify the lobe and the number to identify the hemisphere location. Okay, like if we are saying it is a 32 electrode EEG cap or 16 electrode EEG cap, then each electrode has some name, has some terminology, and it is not randomly named. It the every letter or the number has its significance. Like if we have FTCPO is written on any electrode, then F represent frontal lobe. So the
Ma'am, I think slide is not moving. No, it's stuck at 20-second slide. I think.
standard electrode position,
No, ma'am, no. Now, ma'am.
this how to record EEG signal, you are able to see
Yes, ma'am. Now it is moving.
this.
Oh, yes, ma'am. Now it is moving.
Okay, so this you understood, which I explained, or I should repeat? This was visible last time. This slide was visible last time.
Yes, ma'am. Now it is visible.
So should I explain it again?
Uh,
Okay, I'll repeat. So this is standard electrode position and reference system. It is visible.
Yes, ma'am. Yes, visible.
So I was saying कि okay, so EEG cap which we place on participant's head, it has a standard electrode position system and reference system, because every person has different structure, different dimension of his head. So how should we place the cap so that we should get correct results? So this is the standard systems. It is a standardized placement system of EEG electrode established in 1958 by H. Jesper, a Canadian psychologist. So it is meant to facilitate communication and comparison of results. So this first picture, it is side view of the person, and this is the top view of the person. Okay, so this nasion is the nose area, and this Indian is at the back of his head. Okay, so this is meant to facilitate communication and comparison of the result. So this 10-20, this refers to the distance. This is like a percentage. So this refers to the percentage distance between the adjacent electrode. So that 10% means कि from this area to first electrode, it will be 10% of the total distance, and from the inion to this electrode, last electrode of the occipital lobe, this will be 10%, and then other electrodes will be at the distance of 20% of the total distance. Is it fine? Is it clear till now? Now moving to next slide. This slide is visible.
Yes, ma'am.
Okay, so I have changed the slide. The 10-20 system of head measurement and electrode placement. So you can see this next slide now.
No, ma'am. Not changed.
It is not changed. Why it is not changing?
Now changed.
Okay, so I think I should show this mode only. Is it visible or it is very small?
Ma'am, visible.
Visible. Okay, so let's look like this only. So the 10-20 system of head measurement and electrode placement. So as I explained, each electrode has a specific nomenclature and a specific numbers assigned to the electrodes. Like if electrode is F, T, C, P, O, so it represents their corresponding lobe. Like F means frontal, T means temporal, C means central, P means parietal, and O means occipital. And each electrode has a letter to identify the lobe, and the number will identify the hemisphere. Okay, so number means ki if it is even number, like if I'm saying F2, so the two number is even. So even number will represent the right hemisphere, right side of the brain, and odd number will represent the left side of the brain. Okay, so if I'm saying F3, so it means ki at the left, the frontal part at the left hemisphere. If I'm saying F2, so it means uh, frontal part at the right hemisphere. And Z represent the center part, the center of the brain. So even number 2, 4, 6, 8 refers to electrodes on the right hemisphere, and odd numbers 1, 3, 5, 7 refers to electrodes on the left hemisphere, electrodes along the mid-sagittal plane, which have a subscript of Z. Next slide visible?
No, ma'am. 32. The 10 to 20 system basic steps.
Yes, yes,
Yes,
yes.
ma'am. Visible.
Visible. Okay. And now it is visible?
32. Slide number 32.
Yes, yes. Slide number 32. Okay. So I'm talking about the 10-20 basics system, basic steps. So the step one will be four anatomical landmarks are used to position the electrode. Okay. So you can see over here FP1, FZ, CZ, PZ, O1. So how this CZ is marked? This is center of the brain. This CZ is. So from this nose part to the occipital, the last part, Indian part, slide number 33 visible?
Yes, ma'am, visible, ma'am.
Okay. So you can see from the nose to the Indian part, we will measure the distance of that person. You have in your lab when you will record, you have got your EEG headset. Now you will do some experiment. You will have different subjects. You will call different students to record their EEG, but each student has different head structure. So how will you let them wear that EG cap? How will you do that? So first you have to measure from nose area to the this at the back of the brain. You will measure the total distance. Okay, and the total half of the system and half of the distance you will mark CZ point. Measure over the center line of the scalp from the nasion to the Indian external occipital protuberance, and then you will take 50% of your total. This will be your CZ mark. Okay, you can see slide number 34 here. This CZ will come. So when you are wearing a EG cap, then first you have to place CZ electrode at the center of the brain. After that, you will then start other electrodes. Slide number 35.
Not visible, ma'am. I think 34 did not. Now, yes, ma'am. 34 is visible.
34 is visible, right?
Yes, ma'am.
34. Okay. So we will measure and mark 50% of our total. We have taken this distance. Okay. So we will take midpoint of that distance, half of the total distance. This half distance, CZ, this CZ will be, you will see in your EG cap, there will be CZ electrode will be there. So first you have to place CZ at the center of the brain of the participant, and then you will start placing other electrode. So first you have to set CZ. This is what I'm emphasizing. Ki first you have to mark CZ. Moving to next slide, slide number 35. Then we will take measure and mark 10% from the nasal and 10% from the median, and these are for FPZ and OZ, the other electrode. Prefrontal reference and the occipital reference, OZ. Okay, Z means that the central line. So here will be our FPZ, this will be OZ. And if you will see the side view, then this will be FPZ, this is CZ, and this is OZ. Correct? Now moving to next slide. So this is how after placing this CZ, FPZ, OZ, then you will start placing other electrodes on the participant's head. So then they will be set. So today EG caps are used commonly, and thus placing individual electrode is done only in exception cases. So normally we have EG cap if the person wear. That's the only thing is we have to correctly place CZ, FPZ and CZ and that OZ. This is a picture of my lab where this participant is wearing EG cap. This is gel based. Okay. And here, this slide is visible. Slide number 37.
Yes, ma'am. Visible, ma'am.
Okay. So this here you will see he is wearing EG cap, and here in this screen we record his EEG signal, and what he is watching in the other screen we are showing him the paradigm, the experiment paradigm which we have made. like when he has to close his eye, when he has to open his eye, when he has to perform some task, he has to press some key when he sees some name. So this was the paradigm. So here he is watching his experiment task and performing according to that, and whatever he is doing, corresponding to that, we are recording his brain signal in this system. After recording the EEG signal, then we will see artifacts in the EEG signal. We have a lot of artifacts get induced, different types of artifacts get induced in our EEG signal. So first is physiological artifacts and non-physiological artifacts. Physiological artifacts are like cardiac pulse, respiratory, sweat, eye movement, and muscle and movement artifacts. So if these type of artifacts are there, then we call it as physiological, which are related to this. So you will see the eye movement artifacts, they look like this. If person is blinking his eyes, then we will see this peak. Galvanic skin response over here. If a person is sweating, or if some electrostatic force is there, then we will see this type of. And some electrode, if some electrode has been disconnected or popped up, then you will see no signal in the EEG. It sometimes it get confused that we are getting no artifact, but it is not no artifact. It is no signal is over there because the electrode has popped up. And this is the artifact which get induced in our EEG related to heart rate. When the heart is breathing, we are the heart rate is increased. So these type of respiration, EMG, muscles artifacts, sweating, and this 50, 60 hertz power line interference, these all these artifacts are there in our raw EEG signal. So we have to first denoise our EEG signal, remove these artifacts from our raw EEG signals. And apart from those artifacts, we have some other artifacts also, like cable artifact, photic interference, bad electrode, movement drift, artifact caused by patient being held, EMG jaw clenching, if he's clenching his mouth or anything, then we will see other artifacts also. Even if you are recording your EEG signal in a laptop and you are charging your laptop also through the other cable, then you will see some power line interference in your EEG signal. So while recording EEG signal, you should not charge you should remove other electrical appliances. At least you should not charge your laptop. Otherwise, that electrical signal noise will come in your EEG signal. This is an example of the eye artifacts which get induced in our EEG signals. So since these eye artifacts, we will generally see it when in FP1 and FP2. These are at the prefrontal area, and we are moving our eyes or blinking our eyes. Then the prefrontal area, FP2, FP1, we will get these eye artifacts. You can easily see that because when the person is blinking his eye, then we can see a sudden change in the waveform over here also and over here also. So by just by looking at this waveform, we can remove eye artifacts, but not other artifacts. They are not very much visible. Another method, which is very popular method for denoising our EEG signals, is independent component analysis, which is called as ICA. So what is ICA? Independent component analysis is a computational method used to separate mixed signals into independent components. Like I have a signal which has which get mixed with other signals, then I can do then what we will do, I will divide it into its different independent components, and then I will see okay, this is my noise, then I should subtract those noisy signals from my main raw EEG signals, and I will get clean EEG signal. So this is used in EEG to isolate artifacts like eye blink, muscle noise, power line interference. And for applying ICA, we have to assume, because this ICA is dependent on the method. This whole independent component analysis method is based on the assumption that that if you are applying it on any signal, then the mixed signals which that raw signal is containing, they are statistically independent. If they are not following this criteria, then this method will not be useful in that case. So for EEG signal, we assume the noises which get induced in it, they are statistically independent. That is why we can divide them into independent sources, and we can remove those artifacts from there, isolate those artifacts from our raw EEG. So how ICA works? First of all, we record EEG data with multiple electrodes. Like you have, if we have 64 electrode EEG cap, then we have 64 channels over there across the time domain. So we have recorded that EEG data, and then identifies statistically independent component from mixed EEG signal. As I said, this whole IC algorithm, it is based on the assumption that the mixed components are statistically independent. So it will divide those components into different components which are statistically independent, then allows removal of component corresponding to artifacts while retaining brain signals. And then we can once we have removed those components, then we can reconstruct a clean EEG signal without unwanted noises. So this is the method of independent component analysis. And through you can also understand through this diagram, through this picture. Like suppose in a room we have recorded audio of three singers. So we have, or the person who is speaking, so we have mixed signal. What the ICA will do, this will assume that these are statistically independent, so this will derive their source signal. Like this will distinguish these three signals, and then we will get, like, it is very much useful when we are IC are very much useful in speech processing also. There they use ICA, like for the speaker identification, speaker diarization. These are many applications. Or in a room, many musical instruments are playing. So if you want to distinguish what is the music of harmonium, guitar, and all that, and if we have got the mixed signal, then this ICA can easily separate those individual signals. So similarly, in the case of our EEG also, if we have raw EEG signal and this get mixed with other artifacts, then what it will do, it will divide them into independent components. And once we know, okay, this is my eye artifacts or this is my muscle artifact, then I will remove these things and only mix the main EEG signal and reconstruct my signal, which will be the artifact corrected EEG signal. So this is how independent component analysis works in denoising of EEG signals. This is an example of the raw EEG signal which I had. So I recorded this original EEG signal, and then I applied ICA to this my raw EEG signal. So I had this blink signals, muscle, line noise, EKG, eye movement over there, and I can see their visual also. This you can see in EEG lab or MNE Python also, you can see there also. So, these are the independent components. So I will subtract these signals from my original EEG signal, and this is my corrected uh, EEG signal. Like it has removed the eye movement, EKG, line noises, power line interface. It get induced with the other power sources. If the person is blinking, he's not steady, or he's moving his hand or muscle noises there. So all gets be subtracted from the original, and we will get corrected EEG signals. So as discussed, there are many artifacts in EEG signal. So first, the most common artifacts which we will get in EEG signal, first one is baseline wandering. This baseline wandering occurs at a very low frequency, so they are very slow drift type of artifacts. So they cause slow variation in EEG signals due to respiration, movement, or sweating, and it you will see that appears as a slow fluctuating trends in the signal. And its solution is like you just apply a high pass filter. You put the range like the signal above zero point 0.5 hertz, you pass all those signal and remove the signal those are below zero point 0.5 hertz, then you will just get rid of this baseline wandering type of noises. Then second, the most common noise is power line interference. This occurs at 50 or 60 hertz noise. So for this, we have to just apply a notch filter over here. So these are electrical noises from surrounding power sources. So what we have to do, high amplitude noise at 50 hertz is occur. And so here in India, we at power in our power place we will get 50 hertz noise. So you have to apply a notch filter of 50 hertz, which will remove this interference. And for high frequency noises like the muscles and electrode noises, the muscle noises are generally at a very high frequency occur. So we put a low pass filter, like if the required useful EEG signal which we want to get is from zero point 0.5 to 40 hertz, then we can remove the signal above 40 hertz. So what we will do, we will put a low pass filter and we will say the signal below 40 hertz will be only passed and rest will be rejected. So through this low pass filter, we can remove high frequency noises. For that we can do this hands-on coding for practice. I can show you in the Google Colab how can we apply these things. So the data set which I'll be using over here will be for the uh, is from publicly available data set which I am using, which I will show you in Google Colab. So these data were acquired in the Neuro Mag Vector View system at the GH-MIT Biomedical Imaging Lab. So EEG data from the 60 channel electrode cab was acquired simultaneously with the MEG. They acquired the magnetic signals also. So in this experiment, what was happening, the checkerboard patterns were presented to the subject, to the participants, into the left and right visual field and interspersed by the tones to the left or right ear. And when tone was auditory signal was also given to them. So the interval between the stimuli was 750 milliseconds. So occasionally a smiley face was presented at the center of the visual field, and the subject was asked to press a key with the right index finger as possible after the appearance of the face. So this was the task given to the participant. He when the when he is listening to some auditory sound and a smiley face is coming, then he has to press a key. So this was the task given to him. He has to press the key. So we can see over here. Is this screen visible?
Yes, ma'am.
EEG loading and pre-processing, Python, Google Colab.
Hands-on coding for practice is visible.
You are seeing in the slide.
Yes, ma'am.
Now I'm sharing my Colab screen. It is not
Not
visible?
not shared, ma'am, yet.
Okay. Let me reshare it. Now is it visible?
Yes, ma'am. Easy loading and pre-processing.
Yes, yes, yes. So I'm sharing one simple program where you can see how to load EEG signal and how you can do pre-processing. Pre-processing means you can remove some artifacts from the signals, from the raw EEG signal. So So here we have, this is a Google Colab. So here we have, first of all, we have to import some useful libraries and tools. So here I am using MNE Python library. So first I have to install MNE. So pip install MNE. Through pip install, I will install MNE, and then I am importing MNE. After that, I will load my folder, dataset folder through this command. This data set contains MEG and EG files both. So I have to load all those things, and this raw is equal to MNE IO read. So it will read my input data file through this command. Then I will run it. I have already run this command so that it won't take time, because the installation time is it will take our time. So I've already run this program so that we can save our time. And we, through this print raw info, we can see the information about the raw data which we have uploaded. So this is giving me the full information about the signal. And this is the raw EEG signal, which I have imported into the MNE Python library through that command. Now I have artifacts over here in the signal. So I have to remove those artifacts. So what I will do, as I explained, first, see, I have a EEG data. It can be of 1 hour also. It can be of 30 minutes also. But I will take a certain time period of the data. So I will take first 60 seconds and load it into the memory. So through this command, I will crop my data. So I'm taking maximum time 20. Here I change it. So we can take first 20-second data. So T max is 20 and I'm loading my data. I will crop my whole data to 20 seconds only. And since it is a 60 channel EG, EG electrode recording, but I am taking only, I want to see only few channels only. So here I've selected EG 41 to EG 59. So it will show me only 16 channels only data. So I have taken 16 channel EEG, and now, as I said, first of all we have to remove certain basic artifacts. So my first artifact is baseline wandering, power line interference. This is normally a noise, low frequency noises. So for that, what I have to do, I have to apply a high pass filter to remove baseline wandering, that is low frequency drift. So what I will do, I will apply a filter. I will write its frequency range, like its low frequency range is 0.1, high frequency is nothing, that it will pass all the frequencies and it will pick only EEG data. Okay, so through this I can remove my baseline wandering low frequency noises. And for high frequency noises which are above 40 hertz, which I want to remove, then I will apply a low pass filter. For that, I will take raw filter, low frequency, none, high frequency component 40 hertz. So it will just pass those frequency components only, those signals only which are below 40 hertz, and will block all those high frequency components. So through this command, I can clean my signal from high frequency noises. And for power line interference, like I have a power line interference of 50 hertz. So what I will use a notch filter which is exactly at 50 hertz. So I will use a notch filter written over here, and this I have written its frequency 50 hertz, so it will remove that. Through that, I after this applying all these three filters, then I will plot my EEG signal. So you can see over here, this is my clean EEG signals which I have got after applying those filters. After that, one of the other method is ICA, which I said, independent component analysis. So we can use that also. So in the MNE, this ICA is also there, but we have to separately import it. So for that, what we will use from MNE. MNE we have already installed earlier, but here I'm again rewriting it because I wanted to run it separately. So I installed it separately over here also. But otherwise, if you are running your code in continuation, then you don't need to install it again. So once the MNE is installed, then from MNE dot pre-processing, I will import ICA and this EOG epoch and ECG epochs. Okay, and then I will apply my independent component analysis algorithm to clean my signal. So here through this code, I have given it a, I have first of all, I have input this loaded my input signal. Then here you can see apply ICA to remove artifacts. Then I have to just write ICA is equal to ICA and components 20, random state 42. So this will take my raw EEG signal. This will fit my, I have made a, I have created an instance of ICA through this command, and then I will fit my raw EEG signal into this instant of ICA, and then it will create like in the ICA, we have to give him a standard. We have to give him a sample of the artifacts. Then only it will be able to see. Okay, this type of artifact is there. Then it means this is high artifact. I have to remove this. This is a hard artifact related to heart rate. Then I have to remove this. So I have supplied him a EOG and ECG type of artifact separately. That is also in the MNE Python library only, so that ICA can see it and then will compare its statistical characteristics with the characteristics which are in my EEG, raw EEG signal. Then only it will separate those components, and then it will divide it into different independent components, and then can remove those components. So through that, we have given him a EOG and this heart rate. This EOG is related to blink related artifacts, and then it will remove. It has applied to this through this command, and here we have got EEG data after EOG artifact removal. So through this, we can plot our final clean EEG data. So you can see over here. So this is our clean EEG signal which we have got across different time. I think it is clear till now. We can take a break for 10 minutes. And if anybody has any doubt, then they can ask. Or after break, we can take the doubts. And we can take a break of 10 minutes over here.
Okay, ma'am.
So we will meet at 3:43. Okay, 3:33 is there. So we will meet at 3:43. So let's start.
Yes, yes, please start. Oh,
My screen visible, sir?
no, it's visible. Topo plots in EEG analysis.
Yes, yes, yes. So till now we saw the how to record EEG signal, and after recording EEG signal we get so many artifacts which get induced in our EEG signals. So how can we remove those artifacts from our EEG signal? And we saw one Python program in Colab also, so where we can remove these artifacts through low pass filter, high pass filter, notch filter, and also through independent component analysis. Now, after the removal of artifacts, we will do some EEG analysis. So first we will see topo plots in EEG analysis. What are topo plots? What does it mean? So these topo plots are spatial representation of EEG activity over the scalp, like visual representation or spatial representation of EEG activity, which we have recorded. We can see the certain part of the brain get activated, it is getting high density, high power, and other parts are not that much activated. So through tropo plots, we will get to know this. It provides a heat map of electrical potentials recorded by EEG electrodes, and they are very useful when we want to visualize brain activity patterns and identify region of interest in cognitive and clinical research. So importance, we can see that first of all, the identifying brain activity patterns, it will highlight the active regions during definite cognitive tasks. Like if I have given a task in my one of the project where we were doing behavior analysis, in the case of deception analysis, like when the person was lying or he was saying truth, this, like for that we have lie detectors. But apart from that, through EEG also we can predict whether the person is lying or not. So for those type of tasks, we give him certain tasks, and for those we like to see ki which part of the brain gets activated when we are giving him this task, or when we are giving him for the stress analysis, when we have shown him different videos, suppose to give him stress like horror, we made him watch horror video for 10 seconds, then we have given him some mental task. So these type of task, continuous task he is performing, so he is getting mental stress. So we wanted to see which part of the brain gets activated. So through topo plots, they are very useful. This will be very helpful. And for comparing conditions, also useful in experimental paradigm to see difference in conditions. Like when the person was performing task, then what was his condition? What was the activation in the brain? And when he was in rest condition, then what was the brain activity going on in his brain? Because from experience point of view, I will say, when we are comparing any two person and we want to see how attentive that person is, or if I have designed a game and I am claiming that this through this game that the people become more attentive, then how can I judge? How should I mark it, or how should I give result? How can I say that, rate that? So it should not be absolute marking. It should be relative. Like when the person before using this neurofeedback game or performing this task, how was his EG behaving earlier when he was in rest? And after this task or after this game, now what is, how his EEG is responding, how his brain is responding. So we should always take relative comparison only. Not, we should not give absolute in the case of EEG analysis. And there are also topo plots are useful in artifact detection also. This, they help in identifying noisy channels and or unwanted signals also. For event-related potential mapping, this shows how brain responses and how they evolve over time. And for frequency band analysis, also we can see different distribution of the power in the EEG band in the scalp. We can see okay, which part of the brain gets activated at what frequency. So this will be helpful in that. And how we can interpret those topo plots. So first of all, color mapping. We have warm colors like red and yellow, which indicate higher brain activity, and cool colors like blue, they will indicate lower activity. When we will see topo plots, it will be like heat map. Color map will be there. So if red and yellow dark color is there, then it means this is higher brain activity is going on. So, and also we can see symmetry also. It helps in identifying left and right hemisphere dominance. For the certain type of task, which part of the hemisphere, whether his left brain was more active at that task or the right hemisphere was more dominant at that time. And also changes over time. ERP involved the different regions show varying levels of the activations, which we can see through topo plots. And we have find its many applications like in neuroscience research, understanding cognitive functions, medical diagnostic like identifying epilepsy, brain disorders like this part of the brain, they never get activated when we have given him the star. So means that person might be suffering from there because that part of the brain has not evolved. So this type of analysis helps in medical diagnostics. Then we have brain computer interface using EEG signal for assistive technology also, and for neurofeedback and therapy, so for analyzing the brain states for training the programs. This is one of the example of the project which I did. So I'm taking that example. Like this was the task where that we asked the person to take left and right turn. So it was like intention detection, ki which 90-degree turn he will take. When he is taking left turn, then how his brain is responding, and when he is taking right turn or going straight, then how his brain was responding. So we found different activation through topo plots only, which I'm showing over here. So suppose this is our subject and we made this line. He has to go straight on this line, and then at this line he had to take a right turn. After that, covering this, then he will take again right turn. So when he had actually taken right turn over this point, um, suppose this is t is equal to 0, this t is equal to 0 millisecond. Actually, whenever we do a task, if I'm moving my hand, then my brain has already means make his mind, or brain has make his mind. Okay, so he has already developed that, okay, I have to do this task. So it is actually before actual movement, actual motor movement. So that is why I have given time in minus. Like when the person has taken a turn at 0th millisecond, at this instant, then the brain has already started planning before this turn. Then only it has sent signal to the limb and the person, and then the person has taken right turn. So we plotted the topo plot from minus 450 milliseconds before he has actually taken. So we found that this part of the brain, motor cortex area, this is motor cortex area. So at minus 400 milliseconds, got activated, and then it gave the signal to the occipital lobe. This is the occipital region area. So we can see this area is darker. So from this area, it has given activation to this part. Then it went to this motor cortex area, this of the left hemisphere, and then the this activation part get disappeared. So from minus 400 millisecond to minus 200 millisecond, the brain has developed has already planned. Then now I have to act like this, and then it gave the signals to our muscle signal, EMG signal, and then the person take the turn at 0 millisecond. So we can see at 0 millisecond, actual right turn was taken, As we know, that region of the cerebral cortex involved in motor planning and execution. So this part of the task is related to motor planning and execution. So here approximately 250 millisecond before the actual turn, we can see there is some activation in this region. So from 200, this was the time at when the person has actually taken turn, and this minus 250 millisecond, we can see activation in his brain. So this is just so 200 milliseconds before the actual turn, the we had the activation in our brain region. So we can see this, and these are also different topo plots. This we plotted through the Slorita that tool, and this through the MNE Python library. At the different time sequence, we can see different region got activated. We can see one code over here of the topo plot, which I'll share. Let me share my screen. So this is a Google Colab. I've made one simple program where you can see topo plots, how we can code these topo plots. We can see topo plots of our EEG signal. Is it visible?
Yeah, it's visible.
Okay, so I've taken a EEG signal, and for that I need some libraries. So I will import matplot library, byplot, and NumPy. These are simple these APIs which we have to libraries which we have to import, and we will install MNE to pip install MNE. We will install this, and from MNE library we will import read evoked and some sample. I'm taking sample from the MNE dataset only, which are over there. And so through these are simple prerequisites which we have to use. Then I will load my sample EEG dataset through this command. Okay. So here I have given it an instruction that time for my topo plot will be from 0.05 it is in negative. So 0.05 seconds to zero point 0.1. This is starting time, this is the end time, and this 0.02 is the interval. So 0.05, then 0.07, 0.09, and so on till 0.15 second, it will show me topo plots. So this is the command for that. It is in MNE Python toolbox, which you can easily use. So here I have given some other specifications like bins, how many milliseconds we want to use. So these are, you can change according to you. The main thing was this, which MNE Python topo plot function we should use for this. So this is the main thing. And after, oh, sorry. Then I run that program, and I can see over here my topo plots. So you can see that this is the topo plot at 0.05 second, 0.07 second. The gap is 0.02 second, which I have instructed in my code. So, and this is still 0.15, which I gave, but if we add 0.2, it will exceed. So it is given topo plot till 0.499 seconds. So through these simple programs, we can get our topo plots. This is the another instruction which where I gave the time in minus. So it is giving that topo plot. So according to our task, we can take those epochs, and through those epochs, we can plot our topo plots and can see which part of our brain is getting activated according to our task. So this was till topo plot. Now we will see BCI signal for communication, how we are using BCI signals for communication. So first of all, we should know what are evoked potentials, EP. They are response to presentation of a stimulus. Stimulus means when we have made a paradigm and then we ask subject to perform some task. And when he is performing some task, like in the case of odd ball paradigm, that paradigm. So when he has seen that a different colored ball has come, the odd one has come, and his brain will trigger at that time. So this will be like a evoked potential. We have evoked that potential. So we will see a certain raise in his potentials, in his brain signals. So these are different, and there are different evoked potentials are there, which we usually take and study for our research purpose. So some example of ERP components are like P300 and 100, which are categorized by their amplitude and the latency. Like whenever the P word is a P letter is coming, it means positive, and when N is coming, it means negative. And 300, 100, it represent their time. So P300 is 300 milliseconds. It represent 100, and 100 means negative potential at 100 milliseconds after that stimuli, after that stimulus, which we have given in our paradigm or in the task. And other thing is event related synchronous or desynchronous. They are called ERS or ERD. So some events performing mental tasks such as an arithmetic, object rotation, motor imagery. When we ask the subject to imagine something, like suppose a person, we are doing a neuro rehabilitation task. When a person is not having his hand, but and he has to do motor imagery, means he has to just think that he is moving his hand, although he does not have his hand, but he is imagining. So the prosthetic arm which we have provided him will the brain signal will provide that signal to that prosthetic arm, and the prosthetic arm will then perform accordingly, according to the brain signal. So these type of stimulus, these are studied in the event related synchronous or desynchronous task. This can be utilized for ERD, like when the signal has decreased, desynchronous.
What is the setup for it? Experimental set. I mean, what is the not saying when he's
Sir,
imagine
there
that.
are various setups are there. Like for we make our paradigm, the task we make in PsychoPy tool. You
Okay.
will see that even the brain product, the EG cap from where you have bought, they have their own system also, like E prime Cronus. They provide that also, and in the E primes, their E prime system, you can make your paradigm. But they are, but its cost is 5 lakhs extra, that E prime, they for E prime they charge 5 lakh extra. So if we do not want to spend extra, so what we do, we make our paradigm in PsychoPy. You can see, sir, in PsychoPy we can easily make our paradigm, like eyes closed, eyes open, this task, that task, press key, and then we can connect it through our LSL. You have EEG cap, and this EEG electrodes are be connected to live M. You must be having that white box, which is called as live M. So all the signals get into the live M. The live M has one software which the brain product provide. It is known as LSL. This is for the live streaming of EEG signal. Then this we can connect our psycho by this paradigm through LSL, and we can send those signals to that. So one person can, in the screen, they can see the paradigm, and when he is pressing his keys, then that marker will get correspond to his EEG signals.
So, for example, as you have told that when somebody thinks any particular thing to that region is provoked.
Yes, sir.
So, what would happen? See, for example, when person has means, can we also measure the power of one thought, or when many thoughts are there? Is it possible that
So repeat your question, one thought, or I didn't.
See, for example, as we are told that when we are exclusively focusing upon something, then some region is activated. But madam, when person in a normal condition, when person is not aware, or we say randomly when we check it, so can we know from the mental state of the brain that which type of thoughts are dominant in the mind, positive, negative, and then
Sir, practically speaking, like if I am talking to you, then there might be other thoughts also coming to my mind.
yes, yes,
So,
yes,
yeah. So, like if my phone will ring, then I will look there
yes,
also. So that
yes,
signal
yes,
will also come, and then you will see a huge spike in my EEG signal. So,
yes.
lot of signals and lot of information are there, and our task is this only: ki how we can extract those useful what we require actually from those mixed signals. So that is why we are saying ki eeg is very unpredictable signal. So this is very complicated part of that analyzing eeg signal. So for that only we made certain type of experiment. Like if we if like suppose if I the project which I was doing in for the DRDO. So there they had to design an exosuit which will give assistance to a person to lift a heavy weight. Okay, so there my task was ki I have to think that now I have to lift this box. Apart from this, other thoughts are also coming, but my main thought will be कि I have to pick this box. So we will design this, keeping this motive in the mind. We will design our experiment according to this. So what we did, we design a window screen, and we asked our participants, students, participants in the lab, कि I will show you a picture of a bicep curl like a hand, and when that picture will come, you have to imagine that you have to pick one box and do one bicep curl. And when that picture will gone, you have to then just
Continuously.
keep your hand down. So so that person might be thinking other thing, but this will be his main thought. So this is how we design our experiment, and then we will extract those EEG signal. Okay, if this type of thought is coming, then this type of EEG brain signal will come, these type of evoked ERP will come, and then we will train our model according to that. We will do feature engineering, train our model. So this will be our setup for a ideal situation. Now for real time inferencing, when we are doing actually in the field, so many thoughts will come, but the task will be ki so many thoughts, out of those so many thoughts, so many signals, the our model will try to pick, will try to match that thought which we had implemented in our experiment with this real time thought. And then if it matches, then it will give the output. If it doesn't matches, then it will not give the output. So this is done like that only.
Madam, here one question is there. For example, as you are told that whenever any new thought is coming, there is a spike is there.
Yes.
But madam, here two things are there that some positive thought is there, then also a spike, and some we say unwanted thought, then also a spike. So can we differentiate the from the spike itself that whether the person is boosted up or who?
Means negative thought and
Yes,
positive, what do you
yes,
mean by that, sir?
yes, yes. From the spike itself, are we able to differentiate it? Because
Means from the negative thought and from the positive thought, what do you mean by that?
madam, because at it, both the things are at the thought level. Means something when something is coming, so it's a spike only. What
Sir,
I have understood.
uh, sir, spikes will come. That is why I'm saying that we have to design our marker according to that, experiment according to that only. Okay, sir. Like I will give you one example. Like I was doing stress analysis, whether a person is in stress or not. So
Yes,
if for normal people, I want to check by wearing EEG cap and recording his EEG signal, I want to check whether that person is in stress or not. Okay,
yes,
so this is my main objective. So
yes,
what I will do, first of all, I will design one experiment. I will do some experiment in my lab. I will make whole methodology. Okay, and then I will try to test it that in for the real time. Now how I will design that experiment in my lab for the stress analysis. Why, what I will do, like I will show him some uh, experiment, like for the in our behavior analysis project which I did. So what we did, we showed him some videos, like it was a horror movie was coming, then he saw the horror movie for 10 seconds, then we give him a 10-second of calm video, relax, then we give him a mental task like mathematical formula. We showed him 50 numbers, and out of those 50 numbers he have to just memorize all the primary numbers only. Then he will memorize primary numbers for 10-second screen which is coming. Then in the 10-second relaxation time, then again 10-second I will give him another task. So this is like a stress, he is getting stress while performing those experiments. And I am recording his brain signals also, EEG signal. So I will do my analysis in the lab. Okay, when the person is stressed, he's getting so many mathematical calculation. He's horrified also by looking at the horror movie, horror video, and all that. So how his brain is reacting. So I will record that. I have made a AI model which will say, okay, this is the when this type of EEG signal, then that person is in stress. Now for normal people, for normal testing, I will then record their EEG and I will see whether it is matching with that or not. If it is matching, that is it, then 100% is in stress. So this is how we work.
But from the thin line, that means how do you differentiate that this is leading to stress and this is leading to means not means.
Like sir, in the control environment, the experiment which I did, this was for the control environment. Like I have given him actually stress, because I have given him a mathematical task. He has to do so many mathematical calculations, memorize it also, then press enter also, do that thing. Okay, so he was in stress because he had that pressure. For 10 seconds I have to do this task, memorize also, then I have to do another task. Then simultaneously I am showing him horror clips also. So after go on, this experiment was for 15 minutes long. So in this 15 minutes he was getting so many activities that he got stressed. So this was in controlled environment, and then I will try. But in real time, you are saying, I can understand your problem, because I was also having this doubt, and I discussed with Joshi sir also related to this. Because when we are thinking so many, yeah, we are thinking, so what we can do about that? We are getting so many signals, so many thoughts in a single EEG signal. So what sir suggested me, we can go for invariant space analysis, we can go for linear algebra. So what the invariant space analysis is, that I was doing one work also, I was applying this invariant analysis, but I was not getting results through linear algebra. So I tried this AI model only. In the controlled environment I'm calculating, and then I'm training my model, and then I'm doing. But through pure signal processing like invariant, I have calculated my EEG signal, and I will convert it into invariant space. Like I have 32, if it is 32 electrode EEG signal, so I have 32 dimensions signal. So I will convert, so it is a 32 dimensional space, and in these 32 dimension space I will project each dimension into different Hilbert space. We will match it, we will calculate its principal angle, we will through eigenvectors, and we will see whose value is maximum, whose variance is maximum. So the component which is having high variance means that thought is more prominent out of all those thoughts. While I'm talking to you, I might be looking at my copy, or my mobile, or my wall, or laptop, but my main thing is I'm talking to you. So if you will see all those thoughts, so the main variance, the main component will be I'm talking to you. So its amplitude or its variance will be very high for that. So I should pick only those signal which has a high variance, and then do analysis for that variance. So that might give me the actual thought. But this is just an idea, sir, the idea which we are talking, but actually applying it. So it didn't give me the good result I was applying, but I haven't left it. I will still continue it. But for a project where we have to deliver something or we have to do, so for that we do some experiment, this type of thing, and then train our AI model.
Because main my objective here is that because inside we know that something is either I'm feeling positive or negative, means whether it is encouraging for me or discouraging for me, even when many thoughts are coming. Only my here concern is that whether it is it whether it can be measured at the easy level. Because so
Sir,
that is
a positive thought and negative thought, what do you mean by this? I am not understanding. You are saying about the negative feeling, positive feeling, or the amplitude wise you are talking about?
for example, when we are positive feeling, then the person is likely to take over the many activities. He or she is able to do the means maintaining the efficiency, performing the activities at very well level. But while something is negative, then negative means when something is not very encouraging, then the person finds it difficult, irritating, etc. Then he or she is not likely to maintain the performance at that one. So, there and interestingly, in that case, actually, person is aware of
So
what.
this is attention. This is related to attention, sir. If person is motivated or if he is feeling happy to do that task, so he will be attentive, he will be focused. Otherwise, if he is not that much of interested, he is not feeling good, or he is in low mood, so his attention or his cognitive attention, if I'm talking, that will be low. Okay, so for that we calculate the alpha beta, which I discussed in my earlier slides. Like the alpha region, EEG, we divide the EEG signal from 0 to 40 hertz, we divide into 5 or 6 bands. So alpha delta band corresponds to when the person is sleepy, alpha band and the beta band correspond when the person is highly active. So we can see through that also whether if alpha beta ratio for calculating the attention of a person, many literature are there. So we calculate the power spectral at the alpha band, and then power spectral at the beta band. We take its relative ratio, and if the ratio is high, that means the person is attentive, he wants to do that job. And if that ratio is low, then person is not attentive, is not feeling good, or he does not want to do that task. So these type of analysis are there.
From attention, that is definitely one case is there. Definitely means I can understand your part. That is very easy to distinguish. But sometimes the person is means thoughts are there, but the person is we say even many thoughts are there, but he or she is likely to manage even more things at a good level. Means concentration at a good level.
So, sir, for that case, I would suggest कि then not only EEG will not do the best thing. Then we need multimodal analysis. Like with EEG, we should take other modalities also, like audio, video. Because when I'm doing any experiment, then I should see his face also, his audio, his question answer, his GSR. Like I have done projects where we are doing multimodal analysis, so I can relate that to that also. Like we had one like it was related to deception, like a person is lying or not. So when I will ask question to that person might can say, I have not done this, but his EEG will not lie. His EEG knows कि I'm just saying कि I'm not lying, or I have not done that, but he has done that. So his EEG will be different. His facial expression might be different. Then thermal temperature, the temperature at his skin. So then we if for these type of tasks, many we need to collaborate other modalities like thermal camera, thermal temperature of the skin, audio. It will give it, if cumulative, it will give good results.
Oh, okay. So, Mukund, see, we can have the question. I have to check out that whether they are. You can type the question because as per the EICT Academy, they said that only in the valedictory part it is allowed actually. So either you can type the question, it would be better. In the end, we will take up the question. Yes, ma'am. Yes, Anthony.
Share my results. Okay. So event related potentials. So evoked potentials I covered, I guess, and event related synchronization. So response to a presentation of a stimuli, we calculate evoked potentials, and there are NP 300 and 100 are there. And yes, then we have event related synchronization, ERS, ERD. So some events performing mental tasks such as arithmetic, object rotation, motor imagery modulate the power of the current EEG signal. And this can be utilized for our ERD, decrease of the signal. And neural correlates like N1, P2, N2, N400, P3, and ERN, they are component of event related potentials which we calculate, which are electrical signals in the brain that occur in response to stimuli or cognitive task. Like when a person is stressed and I have given him a certain task and I ask him to press a key, so when he presses the key, then I will calculate these neural correlates N100, P200 means negative potential at 100 millisecond, positive potential at 200, and these things. Then I will get to know about the about his attention, about his neural behavior in the what is going on. So these components, each characterized by a specific latency and scalp distribution, offer insight into different stages of information processing, including sensory perception, attention, and memory. So we have different ERP component. The first one is N1, which is also called as N100. It is a negative going wave that typically peaks around 80 to 120 millisecond after the stimulus onset. It reflects early sensory processing and is often associated with attention. So we can see whether the person is attentive or not. We can see this. We should look calculate his N100. We should measure this. Then we will get to know whether that person is really attentive for that task or not. Similarly, P200. It is a positive going wave that peaks around 100 to 250 millisecond, and it's linked to stimulus specific processing and can be modulated by factors like attention and task demands. Where then we have N200, then N400, a negative going wave that peaks around 400 millisecond, and it is associated with semantic incongruity and is sensitive to violations of expected meaning or context. So for different type of tasks, we have different neural correlates which we should take into account. And P300, a positive going wave, and typically peaks around 300 to 600 milliseconds, and it is related to attention, novelty, and decision making, and is often elicited by infrequent or unexpected stimuli. This is just a video of oddball paradigm which I was talking about, so I will show you here. Here in this, like the odd ball, this is a very famous paradigm, very famous task which we give to our subject. Like he is looking at the screen and a blue color square or ball is coming, and he has to press key when he is seeing the different colored ball. So when he see different colored ball, a stimulus has been given to his brain, and his brain will trigger, and we will get a P300 peak which we will see, that will be we will calculate. So we can see this paradigm over here.
But slide is not moving actually.
Slide is not.
It is 52 event related potential slide is coming.
Oh, I went to if I'm in on 54.
Yes.
Just a second. So this is visible, na? E52.
But 52 only. That is visible for a long time.
Okay. And this
Now,
ERP
now
50.
53. Now it has changed.
Okay. So I discussed this ERP components. This is N1, N100. So a negative going wave that typically peaks around 80 to 120 milliseconds after stimulus onset, and it reflects early sensory processing and is often associated with attention. Similarly, we have P200. This is a positive going wave that peaks around 100 to 250 milliseconds, and it is linked to stimulus specific processing and can be modulated by factors like attention and task demand. When we have some task related to attention, then we will calculate this P200. Similarly, N200, a negative going wave that peaks around 200 to 300 milliseconds, and it's thought to be involved in cognitive processing such as decision making and conflicting resolutions. Then we have N400, a negative going wave that peaks around 400 milliseconds, and it is associated with semantic ambiguity and is sensitive to violation of expected meaning or context. Then we have P300. It is very commonly used. It is a positive going wave that typically peaks around 300 to 600 milliseconds after the stimulus has been given, and it is related to attention, novelty, and decision making. So here I will show a oddball paradigm. So this slide is visible, slide number 54. This oddball paradigm slide 54 is visible. Please tell me, is this slide visible? Slide number 54.
Yes, madam, it is oddball paradigm.
Paradigm, yes, sir.
Yes,
So
yes.
I'm playing a
Cursor is
video. I'm playing a video.
yes. Okay.
So this is a paradigm where blue color is coming and the subject has to respond when he see a sudden change in the color. So whenever the red color has come, he has to press key. So this is like a sudden change. Suddenly he get attentive. So this is a very famous oddball paradigm. Now coming to P300 evoked potentials. I have changed my slide. So this occurred in response to a significant but low probability event, and this is calculated 300 millisecond after the onset, and this was founded in 1965 by
Slide has not changed.
the
Yes.
slide has not changed.
It is. Yes.
Now it is visible now, I guess.
It's P300 evoke potential.
Yes. So evoked potential, I said, it is very commonly we calculate. So it occurs in response to a significance or low probability event. Like in the odd ball paradigm, blue color was coming most probably, and the red coming of red ball was low probability. So whenever it comes, the brain gets attentive, and then the stimulus is given, and he has to press key. So this P300 we calculate at that time. So 300 milliseconds after the onset of the target stimulus. And this was found in 1965 by Sutton and Walter. And this is related to focus specific. When we have something related to focus, then we take this. And there are many experiments related to P300. Or, sir, I have many things to show, and I think we are getting out of, we will get shortage of time. So I should skip this, I think.
Okay.
So I will skip this P300 speller, and I will directly come to this SSVP visual evoked potentials. So a steady state visual evoked potential, this is very useful thing you will see in the literature. This is related to steady state visual evoked potential, and it is a brain response to a repetitive visual stimulus at a specific frequency. Suppose if something is coming at a specific frequency repetitively on the screen, and our eyes are looking at it, then our retina will also start giving information at that frequency. So it will get reciprocated to our occipital lobe. Occipital region is mainly involved with visual when any task is related to visual thing. So this we will see the things in occipital region, and we can we can easily calculate SSVP. So example, flashing light LED is coming at 10 hertz. So this is commonly used in spellers, wheelchair control, or smart home interface. I will show you like this. This slide 60 number 3 is visible.
Like, yes, not visible.
So this is SSV based neurofeedback experiment which we designed. So here what is happening, we designed a neurofeedback neuro rehabilitation, you can say, where in the screen we are showing 10 pictures to the subject. In the one picture it says ki I am hungry. Suppose a person is there who cannot speak, he is physically disabled and he want to communicate. So by this SSVP experiment, he can easily communicate. So what is happening in this experiment? The four pictures are there. One picture represent that I am hungry. The second picture is that I am thirsty. Third picture is that I want to call a person, and fourth is I want to sleep. So if he want to say something out of these four, and he can just look at that picture only. Suppose he is feeling hungry, so he has to focus on this picture, where this picture is that I am hungry. And what is happening in SSVP, that this all these four pictures are flickering at a particular frequency. Suppose this is coming at 10 hertz frequency, means the frames, the picture is coming at 10 hertz. This picture is coming at 12 hertz. This is coming at 14 hertz, or this is coming at 20 hertz. So out of these three, this person can focus on only one subject, on one picture which he wants, that I am hungry. So he just have to look at that picture, and after that the BCI system will get the signal that he wants food. So audio will come that I am hungry, I am hungry, and that person, the other person can give him food. So this was the experiment designed for that. So how this is happening? So this is happening through SSVP. Like this picture, first picture is flickering at a frequency of 10 hertz. He is looking through his eyes. His retina will give this information to his brain. Signal will also start to capturing, will also reciprocating at this 10 hertz frequency. So when we will take this occipital electrode system, like we have O1, O2 and OZ, we have used only 3 electrodes, this 3 signal only, O1, O2 and OZ. Then we will get this information. They will also flicker at 10 hertz, and then we will get to know, okay, this person wanted this thing. If he is focusing on 14 hertz frequency, then occipital frequency will give brain signals that 14 hertz only, and then through that we can get it. So it is a live project which is actually working. So these type of things are helpful in neurofeedback and neuro rehabilitations. So after this, we will be covering multimodal fusion. I think we should take a break for 5 or 10 minutes, and then I will start multimodal fusion. Right, sir?
Yes, no, sure. Okay.
Okay.
So at 4:40 we will meet.
4:40. ठीक है sir.
final vector. Okay. Next.
Let's start, sir.
Sure, sure.
So okay, till now I talked about the EEG signal, only one modality, only one signal. So if we have some experiment where we are handling only one signal, that is EEG signal. What about if we have more than one signal? So this is called as multimodal fusion. When we have more than one signal, like we have audio speech processing. Speech is also non-stationary signal. It also requires advanced digital signal processing algorithm where we have to do processing. We assume it is a non-stationary signal, so we assume that for this while extracting its feature or while processing it, we assume that for this short window that this speech signal is stationary, and we do our Fourier transform for that period, for that interval, and then we do further processing. So and so we, when we have signals other than EEG signal, we do experiment like that also when we have some complicated problems or something. So this is called as multimodal fusion. So the what is multimodal fusion? So it is the process of combining information from multiple modalities. Here modality means signals, so it can be your audio speech signal, video where the data is coming frame by frame, or whether it is GSR is galvanic skin response, how the temperature, how the skin temperature of the skin is, thermal temperature is changing, how the skin is changing, or we have ECG, how the heart rate is changing. um, if we have introduced eye gazing, eye tracking, then how the eye is moving. So all these are different signals, different modalities, and we are combining them. We are doing some experiment where we are taking into account 4 or 5 of them. Then it is called as multimodal fusion. So it is the process of combining information from multiple modalities in order to enhance the understanding or analysis of a certain phenomena or problem. In essence, it involves integrating data from different sources such as text, image, audio, video, sensor readings to gain a more comprehensive and accurate representation of the underlying information. So what are the importance of multimodal fusion? So significance of multimodal fusion lies in the harnessing the unique strength of each individual modality while simultaneously addressing their inherent limitations. Like the sir was asking about the case of deception when the person is lying. So suppose if we cannot through his audio, if we cannot predict or we cannot check whether the person is lying or not, whether he's hiding some information. But through his video, his facial expression, through his EEG, we can predict that. So there are some limitation of some modality and some advantages of some modality. So when we combine them together, then we get some good results. So that is why we do that. In a world where data comes in various forms and formats, so merging information from different modalities will enable us to overcome the limitation of each individual data source. And this is a comprehensive approach, allows us to capture details, patterns, and relationship that might remain hidden when considering only single data in our solutions. So merging of modality is similar to combining puzzle pieces where the whole picture emerges only when all the elements are combined. So this is the importance of multimodal fusion, which we should also look into it. So what are strategies of multimodal fusion? So there is early fusion, late fusion, intermediate fusion, and hybrid fusion. So fusion is when we are like we have recorded signals from EEG, from ECG, from audio, from video, and all that. So how will we fuse them? So there are four strategies through which we fuse them. First one is early fusion. So early fusion is used when modalities are dependent and that is no reasonable conclusion can be made. Means each individual signal, if we look into it and it is not giving us any information, then we combine those modalities. So raw data from different modalities are combined at the input level only, and then they are being fed into the model. Like you will see in the after coming the LLM models, you will see that for predicting any voice or any speaker analysis or any task related to audio, then you will see that the audio is converted into text, and then in the model the audio is also going and the text is also coming, and then the overall result is made according to all these modalities, and then the combined result we get, which is more accurate. So in the case of our signals also, we will do that. So for and extracting features from different medical data sources at various processing level for comprehensive analysis. So early fusion is when we are combining these modalities at the input level only. And intermediate fusion will be when these approaches are combining, data are combining at the intermediate or processing stages. And hybrid will be like when we have combined two three modalities at some stage and one modality at the last stage only. So this will be like hybrid. And late fusion will be at the end only we are combining the results of all these modalities. So this is in detail like early fusion, which I have explained. So I'm skipping this. This is intermediate and hybrid fusion, which I explained. This is late fusion when we have covered the we have from sensor 1 we have extracted feature and applied to a model, and then from sensor 2 and sensor k, and then we are combining their results into data fusion and then giving the output. So this is helpful in identifying the right fusion method, relies on several considerations like data characteristics. So this will depend on data characteristic, task complexity, computation efficiency, and domain knowledge. So this is one of the examples where we have considered multimodal neurophysiological framework for behavior analysis. So this project is for behavior analysis. So here you can see multi signals are there. Like the subject is sitting, he is wearing EEG cap, and in his EEG cap we have ECG, EOG also. So, and eye tracker is over here, and this GSR is here, and from here we are recording his audio and video. So this is multimodal, and we are doing this for his behavior analysis. Okay, so in details you can see over here, this is an activity monitor where we can see his EEG data is being recorded, and here you will see a 28 channel EEG system which he is wearing, 2 channel for EOG for his eye tracking, and this 2 channel is for his ECG. So some 2 channels are placed at his heart, at the in the chest, so to record ECG. For audio video we have used this camera, and for, this is design paradigm which is coming at this screen, and this laptop is showing us multimodal synchronization. Multimodal, by multimodal synchronization we means that there are different modalities are coming and they are coming at different sampling frequency. Like usually EEG comes at 250 sampling at the rate of 250 data samples per second. Our audio signal is coming 16,000 samples per second. So, video 40 frames per second. Then similarly for ECG, for eye tracker, GSR, they all have different sampling frequency. The data is coming at different rates. So we need to have a one synchronization, one time instant, that okay at t is equal to 0, all the signal will start. So will record that. So this system is for that only. And this is the keyboard interaction where the person is pressing his response when he has saw the this paradigm task, and he has to respond. So by pressing the key, he is answering the question or responding. So this task was related to cognitive behavior analysis, and the aim of the work was to employ a multi-model neurophysiological framework for cognitive behavior analysis, and it covers the objective with design, development, and deployment. So we had a neuro method for light detection by EEG brain signal analysis. So this whole setup was for detecting lie detection, whether the person is lying or not. And for that we used six modalities, and this is for his and also this was also for his stressful suspicious behavior detection also. So here we use EEG, EOG, ECG, eye tracking, GSR, audio, and video, and some questionnaire was going on the screen where he had to respond. So this is why this is necessary. So this was for behavior analysis, accurate detection, real-time detection, flexible, adaptable, and robustness and resilient. And its application is when we want to do some mental health assessment of the person, or personalized therapy, stress management, performance enhancement, and human machine interaction. So these are the application where we can use this paradigm. So this was the overall structure. These six modalities, we have time sync data is coming, and we have some event marking, like when he has pressed the question is respondent. So at what time, what he was doing. So this was the event marking, and when then we were doing feature engineering, and after feature engineering, we can do late fusion, early fusion, slow fusion, and then we get the overall our result. So this is overall picture of the whole the whole processing methodology, the overall structure, how this will work. This is another example where we have only used EEG and EMG signal. So here you can see EEG signal is recorded, and here EMG signal is recorded. So similarly, EEG is being recorded, EMG is recorded. Then we will do some pre-processing, then the feature engineering related to spectrogram, short time Fourier transform, or wavelet transform. Then we will train our model, and then we'll detect all that. This is the third used case of multimodal system integration. This is a picture which I'm showing. This is a setup for that project I was talking about. This was for lifting heavy weight. So the person is doing this bicep curl by holding a weight, and this harness is provided for his safety. The treadmill is going on because we wanted our this experiment was for like when the soldiers are running and walking and then lifting box. So for that whole to creating that whole scenario, we have used this treadmill. So person is walking on the treadmill doing bicep curl, means he's holding, we are assuming that he is lifting some weight, and then so what his EG is going on, this other modalities we are recording on these over here. So I will discuss here in detail what is happening over there. So this is for first of all bicep curl with different weight. So this is the screen where we are showing him the paradigm. Like this is a scene, this is a picture of a hand, and person has to move his bicep hand from uh, uh, from 90 degree to this 180 degree, like he is holding something, and then he has to do this bicep curl. So this is the paradigm which we made on the psychopy based task. So here we first of all the experiment starts, we give the beep sound will come with a plus sign will come on the screen, and after 1 second, so this is the relaxation time when the subject gets prepared. Okay, I have to do task. So this is the rest period from 0 to 1 second, and after 1 second we show him this picture of hand where it is the indication that now you have to move your hand, and for we are giving him a 8 second of time where he can do one bicep curl in his space. and after that we are giving him again rest period for 1 second, and then this experiment gets repeated at least for 10 trials. So for 10 trials we record his EEG signal, and we see how his brain is behaving when he is thinking that I have to lift this weight, then how his brain is responding. So, and we have created this task on psychopy based task, and this was used to provide an external cue to start the bicep curl movement, ensuring consistency in the execution of the exercise. So participant performed bicep curls in standing position facing the screen. A proper rest period was given between trials to avoid fatigue and allow for recovery. We should also take into account that the person should not fatigue, he should not be very tired, otherwise his EEG will not be that much accurate. So we should give proper rest time also while performing the experiment. And this is our recorded EEG signal. So these are the markers which we have marked in our EEG signal. So this was like when it was started, then we had trial start, motion end, then again trial start, boundary. So these are different markers which we marked on our EEG signals corresponding to these paradigm. And then we visualize this EEG signal into S-Loretta to show, to see the source localization. So this is, you can see at the 0 millisecond, this part, nothing was there. Then the, from 0 to 60 millisecond, this part, this occipital lobe gets activated. And after 160 milliseconds, you can see this part gets activated. This region is for occipital lobe. This is related to central part and the motor cortex area. So at 230 millisecond, you can see this part is getting activated, and at exactly at 300 millisecond, this whole region gets activated. This is mainly our motor cortex area. So this experiment was related to hand movement, motor movement. So that is why his motor cortex area is activated, and we wanted to see at exactly at what time this is activated. So it is activated at 300 millisecond. So at 0 millisecond, visual stimuli was given to him, and we can see that occipital lobe was activated at 60 millisecond, and motor movement exactly started at 450 milliseconds after the exact when the brain has planned, and then actually he started doing this bicep curl at 400 millisecond after given the stimuli. So this is how we can see how this brain source localization, this S-Loretta toolboxes are, how useful they are. And for the analysis, I did the subband decomposition. And this subband decomposition, you can do any advanced signal processing algorithms, like you can do FDM, frequency domain decomposition, or through analytic signal match filter banks. Also, we can decompose any non-stationary signal into different bands, or through EMD, VMD, empirical mode decomposition, variable mode decomposition. There are several decomposition methods available through which we can decompose our EEG signals. And after decomposing the decomposition of these signals, we can see at these at different bands how the brain was behaving, how the brain was performing. This was the overall methodology of the whole this project, whole work. Like first, we have recorded these, we designed the experiment paradigm, and according to that, we recorded the raw EEG signal. Then we did pre-processing, feature extraction, like DFT based, discrete Fourier transform based, baseline wandering, noise removal, then we did CAR, common average referencing, then ICA based artifact rejection, then amplitude normalization also, then wavelet, we applied DWT, discrete wavelet transform, and spherical harmonic. These are the features which we calculated, and these features are get fed into our AI model, machine learning model. The models are trained, and then we get the predicted trajectory, that how the hand ball is moving in accordance with the EEG signal, corresponding to EEG signal. So these were the results which we found. and that was for fixed phase. Now we studied that when we are changing the weight, like when the person is not wearing, not holding any weight, he's just moving his hands, or when he's holding 2.5 kg weight, 4.5 kg weight, 6.5 kg, 8 kg weight. So when he is holding or lifting different weights, then is his EEG also giving different signal, or it is same? So we found that his EEG signal is also different with respect to the weight. And when we plotted the power spectral density, we can easily see through this plot that when the weight is increasing, his power spectral density is also increasing. So only just by visualizing the power spectral density, we can say, okay, this blue line, he was not holding any weight, so his power was less. When the weight started increasing, then his power spectral density also got started increasing. So this is, beauty of EEG signal processing, advanced signal processing. So we can see what is happening inside and brain according to our motor activity. And this is the ERP analysis also, which we did apart from power spectral density. So we also found that as the weight is increasing, ERP, that event related potential, is also different. It is also increasing. And this is, we plot for different channel, like for PZ, P3, T7, for different channels. These channels are related to motor cortex area for different region, like parietal and the central region, and for the temporal region, the different region, the different ERPs are there, and they are uh, behaving different with different weight, different load when the person is, which the person is lifting. Then we did the classification of the varying weights from the EEG signal. Like we can classify weight through EEG signal. Means we don't know what weight it is only by just looking by the EEG signal. Can we classify them? Then yes, we fed this EEG signal. We use this AI model, Sesame model, which is mainly used for twins with identical weight. So we fed into that, and we found that this model is also able to classify. It is just like a KNN model, so it will cluster these different weights. So we had 5 weights, so 5 different EEG signal was there, so it classified that according to the weights, which we can see over here. Then coming to advanced signal processing, that is a graph signal processing. So what we did, we can do we can do more analysis using graph signal processing also. So here what I did, I made a graph network of my electrodes. So what is happening? I have a GS, total the graph network, which has 3 components. Like V represent the set of nodes. So here I was using 32 channel EEG signal. So 32 node was there, and GS is representing my brain network for any segment S, like for any time period. For suppose for 1 second I'm taking. So for 1 second period, this is my graph network. And ES is the set of edges, which two sets or which two electrodes I am taking for making my graph. So this is set of edges, and WS is the weight of those edges, what I am assigning to those wedge weight matrix. So in our analysis, what we are considering all nodes and edges in the graph at all time instant t. So, and for assigning them weight, we use Pearson correlation coefficient matrix for constructing a W weight matrix. So for that, I did this analysis. This is advanced signal processing. So what is happening over here? A continuous EEG signal is recorded for any time t, and for that I have non-stationary multivariate signals. So what I did, I took a small segment and assume that this signal is stationary in that component. Then I did the eigenvalue decomposition of the correlation matrix, which I calculated for 32 channel EG signal, and then arranged it into the order of their variance values. Okay, eigenvalues and eigencomp. I took the eigenvector and eigenvalue of that EVD and then arrange it into according to their values. Then I took, then I calculated the principal angle of those spaces which I was talking about for the invariant analysis. If we have so many EEGs, if we have so many thoughts, so many signals in our EEG signals only, then how can we distinguish them? Then for that we can do for invariant analysis. So this is so here what we are doing, we are doing principal angle measurement between the two subspaces. So what is this principal angle? So this is a principal angle. This gives information about relative position of the two spaces in Euclidean space. So if we have two spaces F and G and we have set of principal angles between these two subspaces, then what will happen? This will calculate angle theta, which we call principal angle between the two spaces UK and VK, and the maximum angle that will be the R principal angle. So we did it for analysis of our evolution of brain states in our principle through calculating the principal angles. So analyze the evolution of brain states by calculating principal angle between the invariant spaces of consecutive frames, and as the brain remains in the same states, corresponding invariant space of consecutive frames will be overlapping. So so the principal angle between such invariant space will be zero when they are in same space, when there is no change. So the principal angle will be zero, and when they are totally different, we have two different things, then we have a high angle. So did the analysis for our weight. Like I had the EEG signal for 0 kg weight, 2.5 kg. So I found that when I am changing weight, then this principal angle is getting high value. This peak is coming when there is change in the state, because when the person is holding weight for 0 kg, then he is in the same state. Suddenly he is holding a weight of 2.5 kg, then his brain state has been changed. So we can see increase in the principal angle. Then we have elbow angle prediction using invariant space, and then for further we can do characteristic response vector analysis also. So here the same thing is done. First the graph network is formed, and then eigenvalue decomposition is done, eigenvectors are taken, eigenvalue is done, and then what happened, we observe the for prediction of angle, what I did, I observed the equivalent distance between the consecutive subband, that's CRV which I calculated, and found that the whatever will be the angle, whatever will be the distance, that will predict my that will be directly related to my, or will be related to my elbow angle. So these are the results. So this I explained. So what is subband CRV? This is formed using the eigenvectors along with the eigenvalue of connectivity matrix pertaining to particular brain state, which can be given by this formula. So here, if I am using 32 electrode systems and I have, so I will be having 30, if I have 32 electrode EEG signal and 1 second time. So I have calculated its correlation matrix. So the correlation matrix dimension will be 32 cross 32. So the dimension of my space will be now 32 cross 32. So I will calculate its eigenvector and eigen values. So I will get 32 eigenvectors. So out of those 32 eigenvectors, all eigenvectors may not be useful for me. So what I can do, I will pick only the first 4 or the first 10 eigenvectors only. That might be useful for me. So through CRV, I will do that, and then I have plotted these for different weights. So that was related to bicep curl and weight estimation things. And then here I will discuss the lower limb 90-degree turn intention detection. So it was this proposed study was to record the multimodal data from EEG, EMG, and motion capture. So 3 signals were there, EEG and EMG, muscle signals, and motion capture. The motion capture is for recording our kinematic motion, whether the person has turned or he's going. So this kinematic motion can be captured through motion capture IMU systems. So three modalities were taken for this experiment. 10 subjects data were collected, and this was the paradigm which we developed. So person had to go straight. This was 5 meter. Then at this point he had to take right turn. This was around 2.5 meter length. And then again he had to take this turn, right turn. Then he had to go back. Then he will go here, take left turn, and then here also left turn. So this was the overall paradigm, and where we had right turn also, left turn also, and then we wanted to predict his intention. And the person is going, then how his EEG uh, plan, whether that the person will take left turn, right turn, or will go straight. So this was experiment for that. So we did the data recording, use feature engineering, and then we did the source localization in the Florida. And I have discussed this in the previous slide also. So when the person was taking left turn, so this was the time, and when the person took the right turn, then this was his activation in the brain signal. So at 0 millisecond actual right turn was taken. So the planning in the brain was done at minus 250 milliseconds, which we can see over here. So this region is activated. This is our motor cortex area. So this is related to motor movement. So person has taken a turn, and this motor cortex area uh, uh, is having high power spectral density. Heat through this scaling, we can see if it is red, then it is high power, and blue shows the low power area. So in other part of the region, in other parts, we can see low signals. It is not very much activated, but the motor cortex area where the motor movement has to be taken, this part is activated. This was for left turn, and we did the ERP analysis for different frequency bands, also for different delta, theta, alpha, beta, gamma bands, and for different time sequence how the brain was responding. Similarly, it was for right, left turn. And then we did the multi-class classification, that is right, left, and straight, and we got the different accuracy. So the maximum accuracy was 80% only. So actually, any analysis when done through EEG, it is not a easy task. So normally, if we are not doing the proper signal processing, artifacts removing, proper feature extraction, if we are not doing this, then we will not get better results. And when the person is moving, if we are capturing EEG in the case of mobile person, the person is moving, then we get a lot of artifacts. Those artifacts are very difficult to remove. So that is why the experiments which are related to mobile EEG, the accuracy always goes down as compared to when the person is just sitting and he's keeping his head straight. Then we get very nice EEG, clean EEG. So these are the factors which influence our EEG signal. So we need to have a good advanced digital signal processing techniques. Without that, we cannot do any analysis. AI and ML model are useful for certain stage only. They cannot give us a accurate results. They are black box. So for proper analysis, we need to have a good advanced digital signal processing algorithms. So overall, I was discussing about the multimodal fusion. So there are a lot of advantages of it. So here are some prominent advantages. It combines different modalities for deeper understanding of a complex data, and it improves accuracy, reliability in tasks like classification and prediction. It can handle noise variability by leveraging diverse information sources. and it combines evidence to make decisions in ambiguous or conflicting situations. It captures diverse aspects of information, leading to more complete picture. It addresses missing data or complete information by merging modalities, and it opens doors to new application, content generation, and experiences. Thank you. So here I'm done. If you have any questions, then you can ask.
Now it's open to the question.
And if you want, I can show you some paradigm related to stress analysis and deception. I have those also, but let's first cover the questions.
I think meanwhile you can show it, the participants will write the question in between.
Okay.
Paradigms are very important, means interesting to see.
That paradigm, no, sir?
Yes, yes.
Okay. So the my slide is visible right now.
Slide. Yes, MNP, behavior detection.
Behavior detection. So I was discussing about this project where we had to do behavior detection for multimodal analysis. So we had, first of all, we have to check whether the person is in stress or not. So for that analysis, we designed the paradigm. So first it was truth test, then math test, horror video was shown to him, and then calm videos. So this was all these things made one paradigm. And for deception detection, whether the person is lying, is saying truth or not, so for that we made this best friend scenario and mock crime execution. In the deception detection, when the person is lying or not, for that we what we designed, we we asked him, the in the best friend scenario, what we did, we asked him to tell good thing about his best friend. The four scenarios was there. First scenario was he has to talk about his best friend only positive thing which are true. Then second scenario was he had to talk about his best friend negative thing which are true. And then two different situation when he had to talk positive thing about his best friend which is not true, and then negative thing about his friend which is not true. So we had given him a two situation for truth and two situation for lie. So so deception means he will be hiding information or he's lying. In the case of mock crime execution, what we did, we set up a room, and in one box we put something like money, jewelry, and something, some mobile, and we kept the things in one box, and we asked the subject to pick any one of the things. So this was crime execution. Now when he come to the for the experiment, then we will ask him whether you picked something or not. Then if he is lying, then he is recording his EEG, because we had video in that room, so we know whether he has picked something or not. So when he is lying, so how his brain is behaving. When he is saying truth, then how his EEG signal is behaving. So all these are experiment for controlled environment.
Yes,
They are acting it, they are not actual criminal, or they are not actually liars, they are just
correct,
acting it. So but
correct,
for our experiment, for our analysis, we have to design such things only.
correct, correct.
Yes, sir. So for I can show you, just a second. Okay, so for stress paradigm, like this was the paradigm. So here welcome screen will come for 5 seconds and then a neutralist image was given to him. So this is starting we do for the relaxation of the subject. Once he has to relax, he should not be in hurry, he should not be very anxious. Ki, okay, my EEG will be recorded. So first we have to calm down him and then we start our task. Like in the for 3 minutes we gave him a math task, like he had to calculate the palindrome of the number, then the primary number, and all this task was given to him. Then Stroop task test was given. This Stroop test is also very popular test. This is the Stroop, like the screen, um, uh, uh, green, red, this spelling is coming like GREEN. Green is written, but the color of the letter will be red. So he has to see if whether it is right or not. If the green word is coming with green color, then it means it is right. If green word is coming with red color, then it is false. So he has to press key. So this is related to Stuttgart. Then we
So,
gave him a break screen. Then a calm video was shown. Then horror video for 10 seconds was shown. Then calm video, horror video, and this. So through this, we calculated his EEG signal for stress paradigm, and similarly for deception.
so
Now, questions, if anybody
yes,
has
ma'am.
any questions.
So there is a question, ma'am, from participant Krishna. What are the most informative time frequency features you have found for classifying cognitive or emotional states from EEG?
Time frequency feature. Actually, for if you're talking about time frequency analysis, like for the where we have different methods like wavelet decomposition or a spectrogram is there, or we can do short time Fourier transform. So these are for a like if we see these are for signal point of view, but when you're talking about the task point of view, like you have mentioned for emotion, for cognitive, then what we do, we concentrate on the neural correlates, like I mentioned, N100, P300. So these neural correlates, this give the information more correctly about the task related to cognitive and that. But once we have found that, then we have to train our model. But before training our model, we need to have a feature extraction. So for feature extraction, we will use this time frequency analysis methods which are available. And out of all those methods, I will say, like the short time Fourier transform, this is most popular and commonly used method. But from a, signal processing point of view, since we are from signal processing people, so from that point of view, I will say कि short time Fourier transform, they are not very correct methods because they assume the signal to be stationary for that period. So for the accuracy of your method, you have to check whether that signal is actually stationary or not for that period. Okay, like for speech processing, they take signals in 250 millisecond. They take window of 250 millisecond. But in EEG signal, you can see many papers where they have taken 1 second window also, for 2 second window also, and they have applied Fourier transform for 2 second EEG signal, which is not actually correct. That is why I am saying we are signal processing people. That is why we don't appreciate these methods. You can take Fourier transform if you are taking for a very short period of time, like if you are choosing 250 milliseconds, or for 20 milliseconds if you are applying Fourier transform uh, for that window, then it will be correct. So this will be my suggestion. The methods are there, but we have to appropriately choose the time window.
So does it answer your question? In that case, ma'am, is not DWT.
Discrete wavelet transform is also there. Many methods are there. That is why I'm saying that you have to choose appropriately the time window. They are all dependent on time window which you are taking, because these methods are developed for block of data, not for a single bit by bit, means sample by sample data is coming and they are doing feature analysis. They are not made for those uh, type of data. These algorithms are developed for block of data. So this block of data which you are choosing, you have to check whether they are stationary or not. So how you can check whether that your signal is stationary or not? So you can calculate its mean and variance for that period, and then for the and for the next window, and you will see whether it is varying or not. These are statistical characteristics of how we define non-stationary signal. We define any non-stationary signal through its statistical characteristic, like expectations value, mean, variance. So we have to calculate those things, and we have to check if it is not varying for that window, then you can take it as a stationary signal and you can perform your transformations.
Another question is there from Vaishnav. How to identify ERP signals from EEG?
That is what I explained in my slide. ERP signal, these are event related potentials. So what you have a paradigm, you have an experiment, and you ask your subjects to perform a task. So suppose that I have shown that odd ball paradigm, like blue is coming and suddenly the red is coming. So this event has been marked through your paradigm. The person has pressed a key, or you have marked a marker through your psychopy paradigm, and this, so you got this time frame that at t is equal to, suppose 5 second, this marker was came. So you will calculate the ERP, you will take those EEG signals from t is equal to 0 to whatever time you want, 300 or 50. You will chunk those epochs, and then you will average out and then calculate ERP for that. You have many, in MATLAB and in Python, there are functions. You just have to pass your input data and it will calculate your ERP.
Vaishnav, another question from Krishna. Ma'am, when training deep learning models on EEG data, how do you deal with the subject variability and limited data?
Subject variability and
Limited data.
limited data, yes, limited data. For deep learning model, we, the limitation is that we need to have a huge amount of data for that. So for that you can just record more and more data, repeated data. You can increase the number of trials, so that you cannot augment like in other signals, like in that case of text, or you will see many technologies are there, many methods are there where you augment your data. Like in the case of images, if you have one picture of cat straight, then you can increase this data by just sliding it. You can change it, means you can rotate it. You rotate it 10 times and you will get 10 different images. But in the case of EEG, you cannot augment like this. So you cannot play with EEG, because we do not have much knowledge of EEG, how it is actually behaving. So for that we need to record more and more data. Only this is the solution. Or you do such feature engineering, feature analysis, or you reduce your this number of electrodes, because the DL requires this deep learning methods requires the, it is related to your how much feature you are giving and how much data is giving. So if you have 32 channel EEG, you take only 3 channel EEG only, and you divide it into multiple windows. So when you will increase your this time window, you will get so many datas. You take one window as a one data, second window as another data, so you will have a many data. So we deal like that only in deep learning models. And your other doubt was when how we deal with subject variability. So yes, this is the main thing, that subject to subject EEG differs. So we do analysis for subject dependent analysis also, and subject independent analysis also. We and then we, on the basis of that, we generalize it.
Now, how do you basically authenticate the data? For example, is there any medical personnel involved? How do you ensure the threshold or something?
Yes, sir. If we are recording EEG signal, then it is better if some medical person is there because they actually know how to this put EEG cap. But with time, we engineer people also understand how to put this EG cap. That is what I explained. Ki first, we have to measure while this EG cap, how we place this EG cap. So this start from measuring this point to this point. This
Yes.
will be the total distance, and the center of the point will be our CZ point. So we have to keep our EG cap at the first. We have to place this CZ electrode, and then this FZ, OZ, and then rest of then you will stretch your EEG cap. So first these 3 points should be at the appropriate position, and then all the electrodes will be that. So this is what medical people will do. Rest the engineer people will do the EEG analysis recording. So this is the thing.
So one question, for example, as we are measuring basically data for internet addiction.
Internet addiction.
Uh, so, uh, so, so there actually we have to set a level that one EEG responds to the person who may be internet addicted and another may not. So how to draw that demarcating line? Is there any?
First of all, how will you record EEG data? Matlab, what type of experiment you will do that? Because if both,
Actually,
how will you distinguish that?
we have a plan to show the different type of videos, small videos that we have shown in the clips around the
Okay, and
five
if the
minutes.
person is addicted to internet, then what will you do?
See, then definitely what means, what is our assumption that the person who is addicted, then he or she has, means we say, less spikes. That is our assumption. I don't know
No,
whether it
sir, less spike, we cannot say less spike. We can say that, but you can, what I will suggest, you can make a paradigm like that when you are showing a video, then the person who is addicted to internet, he will not get fatigue.
is.
His, because he is used to that.
Yes,
But the person who is not addicted to internet, after 1 hour or after 15 minutes, he will feel fatigue and he will not be interested.
yes,
But that person will be interested and he will be attentive. He will be very much energetic because he
yes,
is used to that.
yes,
And when you, if you are showing him a video very interesting, and if you switch off, you close that, then the person who is addicted, he might feel distress, or he
yes,
might feel that. So these type of changes, these type of stimuli, we can generate in our
yes, very well.
experiment, and then we can, at that time instant, we can see how his brain is reacting, and how the other person who is not addicted, how he is behaving. So from those point of view, then you can compare.
So, madam, how many subject data do we need, minimum do we need to take? Is there any standard that
uh,
5, 6, 10?
Sir, there's no standard, but for, like, if you talk about, if you are using machine learning model, like 10 subject, minimum 10 subject, I will say,
Minimum 10 subject.
minimum 10 subject. And if you are not able to train your machine learning or DL model through 10 subjects only, then what you can use, you can increase this trial time.
Oh,
Like for one subject, if he is doing experiment for 15 minutes, increase it to 1 hour. So you
oh,
will have multiple windows, multiple data, and you use them as a different data points.
oh. Any other question if there's not answered? No question. Thank you. So today I think it is the longest session in a one stretch because on the other days, as a working day, so we have the lunch thing is there. But on Sunday, actually, people want break also, as well as so that's why we put up in the second half and in a one stretch. So they are free in their evening and free in their morning hours. That's why we have to keep this difficult session 4 hour.
Yes,
I'm
people
so
have work
thank you
and
for your
weekends on.
patience because otherwise it is very difficult. 4 hour, but because people want means Sunday means it should be means
Yes, sir.
shouldn't be disturbed.
I
So
had
that is
also many work lined
yes,
up, but I just
yes,
paused
yes.
them. Yes,
Oh,
I
thank
was also thinking that 4 hours is very long, but it was interactive, so it was
yes.
interesting for me also.
No, no,
So,
absolutely for us also.
hmm.
Thank you,
Thank you so much,
madam,
sir.
and all the participants. See, if you have any question, madam, you may type your email.
Email
So,
ID. Okay.
email ID. So if anyone else, they can ask it, not answer. Referring the FTP session. Okay, thank you.
Okay.
Thank you. Thank you.
Okay. Thank you, sir. Thank you
Okay,
so much,
thank you.
sir.